from Indonesia, and best wishes to all of us. Summer course is an experience that will broaden our understanding and help us envision future career paths. We will embark on a series of visits to various natural pharmaceutical companies during this program. The second International Summer Course of Pharmacy serves as a dynamic platform to connect students and professionals from diverse cultural backgrounds. It aims to promote dialogue, exchange of knowledge, and hands-on experience in the exploration and development of natural products-based therapies.
Greeting to audiences, partners, and participants from United States, Malaysia, Philippines, and Indonesia. The Honorable Deputy Rector of Universitas Pancasila, Profesor Dr. Insinyur Arisetyanto Nugroho MM, Insinyur Handrito Harjono MM MPA MSM, and invitation guest, Dean from Faculty of Psychology and Dean from Faculty of Economy and Business. Professor Dr. Apoteker Syamsuddin, Magister Biomed, as the Dean of the Faculty of Farmasi Universitas Pancasila. Professor Dr. Dian Ratih Laksmitawati, Magister Biomed. Dr. Apoteker Gresti Vinotoris Vandini, Magister Pharmacy, as Vice Dean of the Faculty of Pharmacy Universitas Pancasila. And the Honorable Chief Executive of 2nd ISCP UP, Apoteker Gumilar Adi Nugroho S.Pharm, Magister Science Herb. The Honorable Lecturer from Oregon State University, Professor Taifo Mahmud.
The Honorable Lecturer from Kuliah of Pharmacy, International Islamic University Malaysia, Professor Deni Susanti, and Professor Muhammad Tahir Bahtiar.
The Honorable Lecturer from Trinity University of Asia, Ms. Maria Concepcion Sim, Magister Pharmacy RPH.
The Honorable Lecturer from Faculty of Pharmacy, Universitas Pancasila, Professor Dr. Rerenat Apoteker Deni Rahmat, Magister of Science. And finally, all participating students of the 2nd International Summer Course of Pharmacy, Universitas Pancasila of 2025. As the Master of Ceremony, my name is Azumi Sultanika and my partner,
Samai Fadimas.
Representing the committee of this event, we would like to say,
Welcome to the opening ceremony of this year's Summer Course 2025. Now, ladies and gentlemen, before we step up to the next agenda, let's sing together the national anthem of Indonesia, Indonesia Raya, and Hymne Universitas Pancasila. For all participants, please rise. To all the participants, please be seated. Thank you. For our next agenda, we would like to present an amazing traditional dance performance by UKM Besi FFUP. The Nusantara Medley Dance carries the concept of the diversity of regional dance in Indonesia, including North Sumatra, DKI Jakarta, Yogyakarta, South Kalimantan, North Sulawesi, Bali, and West Java. This combination shows unity over existing diversity, no longer a place of division because of differences, but becomes one unit with dominant characteristics. So please welcome UKM Besi FFUP.
Kicir-kicir, ini lagunya, lagu lama Tuan dari Jakarta. Saya menyanyinya Tuan, memang sengaja untuk menghibur hati yang duka.
Hati gembira.
Sore-sore padang bulan, ayak konco pada dolanan. Rene-rene bebarengan, rame-rame daga gojekan. Asal sakatelo ikutan dane opo. Asal sakatelo ati merah dagelo.
Ojo ngono mas, ojo ngono, kan gue janji mas. Kota baru gunungnya Bamega, Bamega umbak menampur di Salakarang, umbak menampur di Salakarang. Pisang silang tanamlah berbaris, berbaris tanam bambu, halaman
Sayang si pato kaang bego gorokan. Sayang si pato kaang bego gorokan. Sakto mangemo tanam aja, mangemo milele lako sayang. Sakto mangemo tanam aja, mangemo milele lako sayang.
Araki kendang di janur kuda kepong, araki kendang di janur kuda kepong. Kopiah sedok
Universitas Pancasila. First of all, I'd like to welcome our distinguished speakers, lecturers, Professor Taher from Kuliah Farmasi International Islamic University, Malaysia. Thank you for coming, sir. And Associate Professor Denny Susanti. Thank you. Professor Taifo Mahmud from Oregon State University, who are joining us from online, I think, from the US. Professor Denny Rahmat, our own professor from pharmacy. Miss Connie Sim, pharmacy from Trinity University of Asia, Philippines. Thank you for coming. And Dr. Meti Funtum from Mahidol University in Thailand. Also, I want to thank the Dean of Fakultas Farmasi Universitas Pancasila, Professor Samsudin, who has arranged this event. I think it's very helpful for our university. And last but not least, I'd like to welcome the participants, 32 students and 5 lecturers from Trinity University. Thank you for coming. And 28 students from Farmasi University Pancasila, who are also joining this summer course. This program is not only a platform for learning pharmaceutical sciences, but also a celebration of international friendships, cultural exchange, interdisciplinary thinking. We believe that initiatives like this will shape our students to become open-minded, globally connected, and ethically responsible future professionals. I think you realize that I don't have a pharmacy background, but I like to give some insights that I think will be valuable for the pharmacy industry in the future. So just bear with me of my thinking. If you don't think it's relevant, then just ignore what I'm saying. As we explore the world of pharmacy, it's crucial to remember that both natural products and chemical medicine share a common goal: to improve health and quality of life. Natural products often come from centuries of traditional knowledge using plants, herbs, and other biological sources. Many modern chemical medicines, in fact, are inspired by these natural compounds, but are refined to ensure precise dosing, stability, and targeted action. However, it's important to recognize that natural does not always mean safer or better, just as chemical does not always mean harmful. Each has its strengths and limitations. Natural products may offer complex mixtures with multiple active components, which can be beneficial but also pose challenges for standardization, quality control. On the other hand, synthetic medicines can be more consistent, faster acting, and more thoroughly tested through rigorous clinical trials. The real opportunity for pharmacy lies in combining those wisdoms of nature with the precision of modern science. By understanding both, pharmacists can guide patients to make informed choices, whether it's using herbal remedies responsibly or ensuring safe using synthetic drugs. So as professionals, let's not see this as a competition between natural and chemical, but as a collaboration for better health outcome. So to all participants, we hope you all enjoy this lecture and discussions and cross-cultural interaction during the course. May this experience leave lasting impact on your academic journey and professional development. For the organizing committee, I'd like to thank Farmasi University for your commitment and understanding and the preparation that make this event possible. With that, With great pleasure, I hereby declare the second International Summer Course in Pharmacy officially open. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Mr. Handrito, for your wonderful speech. Now we would like to invite Mr. Handrito. We would like to invite you to strike the gong.
Okay, for documentation committee, please proceed to take the picture. Okay, Mr. Handrito, you're pleased to strike the gong in 3, 2, 1. Please strike the gong.
For our next agenda, we will be taking picture with our honorable guest, Professor Dr. Insinyur Arisetianto Nugroho. Please come up to the stage. Professor Dr. Apotheker Samsudin Magister Biomed, please come up to the stage. Professor Dr. Dian Ranti, please come up to the stage. The documentation committee, please take picture in 3, 2, 1.
Okay, now please take a picture with Salam Pancasila. Okay, that's good. Okay, please proceed the picture in 3, 2, 1. Okay, now for the second photo, we are facing the banner in front of the stage. Yeah, for the documentation committee, please go up to the stage. Okay, please prepare your best smile for this photo.
Facing the banner.
Okay. Okay, ready? Please take the picture in 3, 2, 1. Okay, now give your best smile in 3, 2, 1. Now with Salam Pancasila. Ready? In 3, 2, 1. Okay, thank you. Now you may be seated. Thank you. Okay, so for our next agenda, there will be material will be given by Professor Taifo Mahmud from Oregon State University. Professor Taifo Mahmud is a professor of pharmaceutical science at Oregon State University with a strong background in natural products and medicinal chemistry. Holding a BSc and apothecary degree from North Sumatra University and Magister Science and PhD degrees from Osaka University. His extensive professional experiences includes roles as a research assistant professor at the University of Washington, a faculty member at OSU, and a co-founder of Gadusol Laboratories. He has been recognized with numerous honors and awards, including the American Society of Pharmacognosy Nation Investigator Achievement Award. Dr. Mahmud research focused on the biosynthesis of natural products, as evidenced by his extensive publication record of 132 peer-reviewed articles, a Google Scholar Edge index of 44, and 10 patent applications, including 5 US patents and 1 Russian patent. His work includes significant contributions to understanding the biosynthesis of compounds like acarbose, polydamycins, and pactamycins, and developing engineered production methods for various natural products. So we are honored to have Professor Taifo Mahmood from Oregon State University, who will be delivering today's lecture on engineering natural pharmacy, advance in natural product biosynthesis for future medicines. So Professor Taifo Mahmood, the floor is yours.
Can you hear me? You can?
Yes, we can hear.
All right. All right. Thank you very much for the introduction. Good morning, everyone. It is my pleasure to be able to join you today to give my lecture on biosynthetic engineering or engineering nature pharmacy and the use of biosynthetic approaches to make new drugs. So let me share my slides. Can you see my slide? All right. So as introduced, my name is Taifo Mahmood from Department of Pharmaceutical Sciences, Oregon State University in the United States. So today I would like to talk about how we engineer organisms that produce natural products to get new compounds or bioactive compounds that have potential to be used as drugs or medicines. All right. So, of course, many of you know that common therapeutic agents that are used currently in clinics, they derive or they come from nature. Also, they are synthesized chemically sometime, and more we see a lot of biologics being used, which is biological macromolecules like monoclonal antibodies or growth factors and so on. But historically, natural products or compounds that are isolated or produced in nature have been used as medicine. So traditionally, people use herbal medicines, right, like herbs and plants products. And later on, we know that some animals also produce some kind of toxin or, peptides that can be used as medicines. And then fungi or fungus can produce medicine like penicillin, beta-lactam antibiotics, and cephalosporins. And then bacteria also have been known to be a prolific source of natural products, like, they produce compounds like tetracycline, chloramphenicol, erythromycin, etc. So, in fact, if we look at the new drugs, small molecules that have been approved by the FDA, Food Drug Administration here in the US from 1981 to 2019. So about 1/3 of those drugs or small molecules, they came from nature or derivatives of natural products. So botanicals is not that much, but natural product, purified or isolated compounds is about 5%, but a lot of derivatives of natural products have been approved as drugs. There are synthetic compounds, like 1/3 of them, even though they are synthesized chemically, but either they contains natural product pharmacophores or they are inspired, the structure are inspired by the structure of natural products. So about only 1/3 of those drugs really synthesized chemically without any correlation to natural products. So obviously natural product continue to be an important source of new drug leads. But as currently we still need a lot of new drugs because many bacteria become resistant to the existing antibiotics. So we continue to look for new medicine or new bioactive compounds that can treat bacterial infection, fungal infection, viral infection, treat cancer, etc., etc. Unfortunately, the traditional approach to natural product drug discovery is no longer sufficient to provide novel compounds. So if you go out there and try to isolate compounds from plants or animals or bacteria, it is more and more difficult to find new bioactive compounds, mostly because many of them have been isolated or have been identified previously, so you don't really get new compounds. But scientists then try to solve this problem by finding natural products from new sources like organisms from extreme and unique ecosystem, for example, looking for secondary metabolites or bioactive compounds from symbionts, plant symbionts, or animals symbionts, microbiomes, for example. But we also have a lot of advanced technology, cutting edge technologies like new tools, new instrumentation, new technique, high throughput screening, etc., to to basically to make this process easier and faster. And then also people develop new approaches like contemporary approaches such as biosynthetic engineering, engineering of nature, medicine production, for example. So these contemporary approaches, actually many different kinds. One of them, what we call biosynthetic engineering, basically we engineer the biosynthesis pathway of natural products to make novel compounds or to make new compounds. We can use chemoenzymatic approach, combination between chemistry and enzymology or biochemistry to make new compounds that way. And then because more recently the advance of biotechnology and DNA sequencing, gene sequencing or genome sequencing that provide a lot of new information about how organism, they contain or they have a lot of potential to produce natural products, but unfortunately under normal condition or laboratory condition, sometimes they don't really produce the compound that they're supposed to be able to produce. So now we can try to mine those information and try to identify natural products that way. And then synthetic biology, this is a more advanced approach that we generate or create new organism to be able to do something that they are not usually do. So we can synthesize certain products that we want in a biological system. So today, as due to the limited time that I have, I will focus my talk on biosynthetic engineering and synthetic biology. Hopefully, this will give you some perspective or some information about how far we have been able to get in the field of natural product biosynthesis and related to the production of new medicine. So when we talk about natural product biosynthesis, but as you can see from, just the word biosynthesis, basically still require a production or synthesis of a molecule, but occur in the biological system. So basically, all secondary metabolite or natural products need to be synthesized, like what people do in the lab, but all those processes occur in the cell or biological system. So, of course, to do that process require enzyme or proteins, right, that can catalyze the reaction. And of course, enzyme or proteins come from RNA, which is originated from DNA. So this kind of a central dogma of biochemistry that everybody should know or already know, from your biochemistry class. So organism have chromosome, which consists of DNA, which is the blueprints of everything that occur in the cell. That DNA will be transcribed to RNA and then translated to protein, or in the case of biosynthesis, is called enzyme. So consider, like bacteria in that single cell, small organism contain or consists of a lot of machineries in that cell, like a small factory that can produce natural products. So many of them really just looks like an assembly line, in the production of cars or motorcycles or other products, right? So example of this assembly line, like natural product biosynthesis, for example, is catalyzed by enzyme called polyketide synthesis or non-ribosomal peptide synthesis. So from the name, you know that this enzyme catalyze the production of polyketide compounds. This enzyme produce a peptide, peptide compounds. And of course, many other compounds like terpenes require terpene synthesis. Aminoglycoside antibiotics, for example, require a set of enzyme that catalyze the reaction to make those type of compounds. So again, due to the limited time, I won't explain too many different system. But one example really that I would like to focus today is polyketide synthesis, or known as PKS. This is really a mega enzyme, or um, you know enzyme that the size is very big compared to regular enzyme. It's similar to fatty acid synthesis, basically enzyme that synthesize fatty acids. But in the case of polyketide synthesis, it produce polyketide natural products. This type of enzyme, they are multi-domain. So this I label them with different names here. So this is the acyl transferase domain, ketosynthase domain, acyl carrier protein domain, dehydratase, and ketoreductase. So one big enzyme, but they have they have the capability to catalyze several different chemical reaction. All right, and this kind of enzyme use small molecules like acetyl CoA or malonyl CoA to as building blocks to produce polyketide compounds. For example, erythromycin or rifamycin, for example, those are polyketide compounds. And this type of enzyme are divided into several different types. Type 1 polyketide synthesis is called modular polyketide synthesis or iterative polyketide synthesis. For example, they are involved in the production of erythromycin, lovastatin, for example. Those are made by type 1 polyketide synthesis. And then type 2 polyketide synthesis slightly different, but have very similar principle. Produce doxorubicin, for example, and type 3 polyketide synthase produce flavonoids, chalcones, and so on. So this type of enzyme, they are ubiquitous in nature. You can find them, in plants as well as in bacteria and fungus, in lichens. They are very important for the production of natural products because many, many natural products or bioactive compounds, including the one that are used in clinics, they are derived from this type of enzyme. So just very quick to show you the principle or the mechanism of this type of enzyme. So basically, this enzyme, as I mentioned, they contains a lot of different domains. One big enzyme has multiple functions. So there's a place, or the first one is called loading domain. So anything that is used as a starter unit will be loaded to this enzyme attached to this acyl carrier proteins. And then this acyl transferase domain really is the one that recognize what extension units to be used to extend this molecule to make the polyketide compound. For example, this ketosynthase uh, will take this acyl carrier protein first, transfer to module 1, and then this acyl transferase enzyme, for example, will recognize methylmalonyl CoA as substrate, so it can select specifically what extender unit to be used, and then take it and put it on this domain, and then transfer it to the acyl carrier protein of module 1. And then this is reaction between this extender unit with the loading or the starter unit here occur through the decarboxylation and then the nucleophilic attack here to give these products. Right, so in the enzyme, as I mentioned, there is a ketoreductase and dehydratase domain. Ketoreductase mean this enzyme reduce ketone here to alcohol, to OH. And then dehydratase basically remove the OH to make a double bond. So this compound being basically bring to the ketoreductase and dehydratase to be modified and then to get the products that it wanted. So you have this double bond. And then the product of module 1 then is being transferred to module 2, and the same process occur here. This acyl transferase domain also need to select what kind of extender unit to be used. So this is specific for malonyl CoA, for example, so that it can be taken up and then transfer. And the same thing occur again with only ketoreductase present. There is no dehydratase, so that you will get a hydroxy group as a product here instead of double bond, for example. So this happen again and again until you have long polyketide chains, and then this thioesterase at the end will be able to cleave the products, whether to cyclize it or you will get a linear compounds. So to make such complex molecule like rifamycin B here, for example, really require a lot of complex enzyme with all this domain with different activities, really extending this starter unit here, which is 3-amino 5-hydroxybenzoic acid, to make this long chain polyketide, which is then being cyclized to make cyclic backbone like this. And then there are a number of tailoring genes that make tailoring enzyme that would decorate the products by adding acetyl group, methyl group, for example, and some other modification of the molecule. So that's the way how bioactive compound is produced in nature or in biological system. So about this rifamycin B, the one that I just mentioned, actually this is very important compound because this is a precursor of rifampicin. As you know, rifampicin is a first-line anti-TB drugs, right? A first-line drug to treat tuberculosis. It is a polyketide natural products. It is a potent inhibitor of bacterial RNA polymerase. So it's inhibit RNA polymerases in bacteria, meaning it block the production of RNA in bacteria. That's the way how rifampicin kill bacteria. But unfortunately, nowadays more and more, Mycobacterium tuberculosis, the bacteria that cause TB, become resistant to these antibiotics. That we call them multi-drug resistant, MDR TB or XDR TB. This is extensively drug resistant tuberculosis. All of them are resistant to rifampicin. But if you look at the mechanism of rifampicin resistance, we'll be able to see based on this calculation or modeling done by Molotskov and et al. So basically, when they dock rifampicin to the wild type RNA polymerase enzyme, which is sensitive to rifampicin, they can see this compound can easily get into the pocket of this enzyme that resulted in inhibition of the activity of the enzyme. But when the enzyme mutate itself, they become resistant. For example, in this case, mutation from histidine to tyrosine here, so the IC50 jumped from 0.012 to 1,130. So it is significantly, height jump, meaning the bacteria become resistant. So when they try to dock this structure to this pocket, you can see here the modification of the enzyme has make the pocket become smaller, so the compound cannot, get into that pocket anymore. So of course you can try to find completely different drugs, or you can try to modify this molecule so that the conformation change does not basically interact or get bumps directly to this enzyme. So one possibility really to modify the backbone of these polyketide chains of rifamycin, but of course this is very difficult to do chemically. So that's where we tried biosynthetic engineering approach in this case. Of course, you can modify this backbone, whether by removing this methyl group, for example, to make unmethylated, or desmethyl version like this, or remove this methyl group, anywhere you want. But then due to the knowledge, or because we know how this molecule is made, then we can go back to the biosynthetic the biosynthetic pathway, and then try to engineer it, right? So like if you want to modify your car, for example, if you know how the car is made, you can remove one section of the car and so on and so forth. So in this case, we replace the AT domain of module 6 here that introduce or responsible to add this functional moiety with the AT domain that recognize malonyl CoA. So instead of this unit being added, now we add this acetyl unit. So that's the product at the end really does not contain this methyl group at this position. So this is a really a nice approach, because otherwise this cannot be done by chemically, by selectively only cutting 1 cc bond at this position to make this molecule. Of course, this molecule can be then modified just when people modify rifamycin B to make rifampicin. So this requires 4-step chemical reaction, and we are able to make this 24-desmethyl rifampicin, a modified version of rifampicin. And indeed, this compound is more active against rifamycin or rifampicin resistant Mycobacterium tuberculosis. So this is kind of to give you some general idea as to how we do all this thing. For example, well, we started with genetically established organism, bacteria, for example, or fungus that have genetic information established, like genome sequencing data and so on. And then you can engineer that organisms, for example, to make some mutants depending on what you need. And then, of course, then you can optimize the culture condition to produce the compound that you are expected. And this can be analyzed using metabolomic approach, using LC-MS/MS, for example. And if there is a interesting compound to be isolated, then you can do isolation procedure by using HPLC or several other chromatography, and then do structure characterization by NMR, nuclear magnetic resonance, and mass spec, and UV, IR, alpha D, and others. And then you can also test the compounds again different bioassays, like antibacterial, anti-cancer, and so on. Really depend on what the target that you want. So sometimes people can use this bioassay earlier as a guide, like it's called bioassay guided isolation, just to find the compounds that is active against certain organism or certain function. Yeah. Right. So maybe some of you wonder, so how do we do this biosynthetic engineering, right? So there are many different ways. So you can knock out the gene. So gene knockout is a very common or commonly used genetic engineering technique. It's used to disable a specific gene in the organism. You can introduce mutation or disrupt the gene completely, so that you basically kill the activity of the genes or the protein. So that way you can control, or you can learn actually the function, what the function of that gene, for example. You can also swap the genes in the genome with different genes or different DNA construct that you produce. That way also you can get, depend on the purpose of your research. And then more recently, maybe some of you or many of you have heard about the term of gene editing, right? That's a technique that people can precisely and specifically modify the DNA, using CRISPR-Cas9, for example, to to engineer or even to fix certain genes in the cell. So that can be used also to fix some genetic disease, for example, right? So what happened is when you construct a plasmid and introduce into one cell, then, well, depending on the construct, for example, in this case, if you want to remove this green DNA or genes in the chromosome, right, you can clone this DNA fragment upstream of this green one, and then downstream of the green one. So you get this yellow and red DNA, you put into the plasmid and you transfer it into the organism. And then this is a natural phenomenon that we call homologous recombination. If they are exactly the same, they tend to recombine or reorganize. And then the result of that is that you introduce the whole plasmid into the chromosome like this. So this is the product of the first crossover, right? And then now you see this red DNA and this red DNA, they are identical or homologous, you can say. So sometimes you get this double crossover, this homology or homologous sequence then will do homologous recombination again. The product of that, then you get this one go down here to here. So you get this mutant chromosome, and then you get the plasmid with this green DNA inserted in there. So you can see the wild type chromosome contain this gene, but the mutant chromosome is no longer contain that genes. That's the way how we remove certain gene or we knock out certain gene. All right, then you can swap this with other gene as well if you introduce that genes in this plasmid. So this is what we do. This is a different project that we have done in the lab about biosynthetic engineering of pactamycin. This is another very complex molecules with this five-member ring structure, with all this functional group attached to it. It's very difficult to synthesize molecule. And then the first thing, of course, we have to identify the biosynthetic gene cluster. That's the cluster of genes that are responsible to make these compounds, right? That's the first thing that you need to do. If you want to engineer natural products, of course, first you need to know how that compound is made. So the first thing, look at the genes. Come back to this central dogma of biochemistry. From the genes, you expect to get mRNA and produce protein or enzyme that catalyze the reaction. So as you can see, there are so many genes that are involved in making this complex molecule. So we have spent a lot of time really to understand how this complex molecule is made. So each of these circle here is represent enzyme or proteins, right? So many proteins involved in constructing this molecule from the starter unit called 3-aminobenzoic acid here to make these complex molecules. So the enzyme that I show here is the one that decorate this backbone or this core structure of pactamycin. So one of these enzyme add the side chains here, and the other enzyme add this methyl group here, and so on and so forth. So we have been able really to engineer strain of bacteria that produce this compound by knocking out or inactivating several of these enzymes, and we are able to produce compounds that are normally not produced by this bacteria. For example, several intermediates, and even we could add alternative substrate, is fluorinated compound, for example, to make a fluorinated version of this molecule. So as you can see, uh, using this biosynthetic pathway engineering, you pretty much can make many different compounds that typically nature do not produce. Some of them really give very good biological properties. For example, the parent compound pactamycin itself indeed is very active compounds, has very strong antibacterial activity, strong antimalarial activity, but very toxic, right? So this is not a good compound to be used as drugs. But by modifying the structure using genetic engineering or biosynthetic engineering, for example, by removing this, side chains to make this compound called TM026, and suddenly this compound very selective against malarial parasite, Plasmodium falciparum, but does not have antibacterial activity and 30 times less toxic compared to the parent compound. So this is obviously very promising because we can make the compound very specific against certain, infectious agent, in this case, Plasmodium falciparum. The same thing with STM25 that we modified at two places here, also retain antimalarial activity, but does not have antibacterial and cytotoxicity anymore. So this is something that has potential to be developed further as a new antimalarial drugs. Now, the remaining of my talk, I will very quickly talk about synthetic biology in natural product discovery. The definition of synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. So people use synthetic biology approach to make biofuel, for example, to make some other materials using biological system. For us in the field of pharmacy or pharmaceutical sciences, of course, we tend to use this technique or this methodology to produce bioactive compounds, in this case, natural products. So some people or scientists have used this system really to produce artemisinin, for example. Artemisinin is a new, well, not new anymore, but this is an anti-malarial drugs, right? Like artemether, arteether come from artemisinin, which is produced by plant, Artemisia annua. But of course, the production of this compound in plants caused some dilemma because you need to have very big field to produce or to grow all these plants and to produce this compound. But then sometime when the farmer that grow all these plants produce a lot of them, a lot of it, then the price drop and they don't make money. And then when the next year, they decided not to grow the plants, and then this compound become limited, and then the price goes up. So basically, it causes a lot of headaches. The price ups and downs all the time. So one solution perhaps is to produce the compound in a different system. So of course, you need to know how this compound is made in plants first, or how it's synthesized in plants, and then use those, the genes that are involved in making those, making that compounds, and then engineer them and put them into yeast, for example, like Saccharomyces, and then through fermentation, then you can feed the yeast with sugar or ethanol to produce, well, so far, you know, only the intermediate of the compound can be produced, which is then modified chemically to get the final products. But it is a feasible approach because they can make quite high yield of the compound or the intermediate uh, to be used for further modification to make this active compounds. So other lab also have tried to engineer yeast to produce morphine or yeast to produce vinca alkaloid like vincristine and vinblastine, for example, right? So in the future, then perhaps we don't really need a lot of big space or lands to grow all these plants to get bioactive compounds, but all can be done in a in a fermenter or by fermentation in the factories, right? So, but that require a lot of studies, a lot of knowledge of how those molecules are made originally, and then we can transfer those genes into a different system. That's what we call synthetic biology, right? You engineer organism to be able to do something extra for us. So, for example, in my lab, so many years ago, we found that fish actually can produce sunscreen compounds. So fish has genes, actually only two genes in the fish that can make a small molecule called gadusol. But this compound is very unique because this small molecule can absorb UV at around 294 nanometers. So fish made this compound to protect their eggs and embryos from UV radiation. So fish that live deep down in the ocean, in cave, for example, they do not have these genes. So of course, the problem with sunscreen, the one that being used nowadays is synthetically made sunscreen compounds. They cause some problem environmentally because those synthetic sunscreen compound can kill coral, can cause coral bleaching and kill coral embryos. So some places like Hawaii, for example, have banned certain type of synthetic sunscreen. So there are a lot of interest of developing natural sunscreen. So of course, isolating this compound from fish is not feasible. So what we did was then we cloned these two genes from fish and then transfer them into yeast as well as into bacteria called Streptomyces silicolor, and we are able to produce this molecule by fermentation. So this compound is currently being developed commercially by a company. Hopefully, one day this can be used as alternative or as natural sunscreen products, right? And what we did also further really to try to make new compounds that are not naturally made in nature. So based on the knowledge that we gain from the function of genes, from different sources, we are able really to take genes from fish, for example, and one gene from soil bacterium, and two genes from plant pathogens, for example. Of course, we know their function and try to put them together. So it's kind of genetic mix and match, and then try to induce these genes to make enzyme that can catalyze a series of chemical reaction to make a products. And we are able really to produce molecule like this we call gadosporins, A to C. These compounds are not produced in nature. So you see the technology has advanced so far so that we can really create natural compounds or unnatural compound naturally in biological system like bacteria or yeast that hopefully has certain function. In this case, this gadosporin A and C absorb UV around 340 nanometer, which is the UVA range of UV. So if you recall, gadusol protect us from UVB, and this molecule protect us from UVA, right? So it has a set of compounds that can be used as sunscreen compounds or sunscreen products that protect us from both UVA and UVB. But all this seems very, very straightforward, but actually that's not really the case, because in biological system there are a lot of other factors that involve. So this is called, regulation, right? So this only one slide. I just want to remind you that biosynthesis of natural product in a biological system actually is very tightly regulated, right? So there are several regulatory pathway. It's called global regulation, as well as pathway-specific regulation that regulate this the production of natural products. That's the reason why sometimes, depending on the condition, some natural products are not being produced by the organism because of this old regulation. So this is another field that I think very interesting or very important to study, so that we are able to control, or we are able to make sure that the regulation really positively impacting the production of the bioactive compound, like antibiotics or anti-cancer drug and so on, right? So with that, in summary, I would, emphasize that natural products continue to be an important source of new drug leads. Cutting-edge technology and genetic-based approaches have changed the way how drug discovery and development research is conducted. I've introduced to you by synthetic pathway engineering and synthetic biology. Those have opened up new opportunities for discovering and developing new drugs. And we have like genome minings, which I don't have time to talk today, but that's another approach that has been used more recently to look for new natural products. And more importantly, really, to be able to do all those things, we need to understand how natural products are synthesized in nature and how they are regulated, so that we can be successful in discovering and developing new bioactive compound using this biotechnology approach that which are biosynthetic pathway engineering and so on. With that, I would like to stop here, and I'm happy to answer any question if you have one. Thank you.
Prof. Taifo Mahmud, any question?
For the communities. My name is Veronique. I'm from Pancasila University. Thank you, Professor Taifo Mahmud, for the lecture. It's really inspiring. I want to have some questions. So first of all, how do we actually know if the compound is modifiable? Like if you know that it's compatible to modify it, and then like the mix and match gene mix and match, yeah. And like what are the biggest obstacle for the modification research. And maybe from Professor Taiful Mahmud, like what do you think that the young research need to take to improve the drug discovery? Maybe that's all.
Right. Can I answer directly?
Yes, please.
All right. All right. So that's a very good question, right? So as I mentioned just now, everything looks smooth and looks straightforward, but actually there are a lot of things that we need to optimize, a lot of things that we need to understand, right? So in the case of biosynthetic engineering, one aspect that we have to do first, kind of the prerequisite of the research, really to understand these the biosynthetic pathway, understand the function of the genes, understand the function of the enzyme, and so on and so forth. So that's kind of more basic science. So that's why some people misunderstood or don't understand the importance of basic science, right? To understand those biological or biochemical function of genes and enzymes. So once you know those functions, then and you can figure out, for example, the biosynthetic pathway. Based on that knowledge, then you can design, you have a hypothesis that, for example, if you modify the system this way, presumably you will get, this type of compounds. If you modify this system that way, you get different compounds, for example, right? So, of course, that need to be done, to be tried. Sometimes if you're not lucky, you don't get what you want. But if you are lucky and your hypothesis is correct, then you will be able to get the products that you want. So, well, we have the luxury being in academia, to play around and do a lot of basic research, try to understand all those things. And obviously, if you look at the biosynthetic pathway of pactamycin just now, I show a lot of enzyme involved. There are many very, very interesting enzymes that are novel and have not been published before. So, for many years we have been able to publish many of those interesting enzymes. So from academic standpoint or scientific standpoint, actually there are a lot of exciting things happen. But then from the application of it, of course, really depend, if what we hypothesize is correct, and then you get a product. In case of synthetic biology, mix and match, of course, that's the same thing. You need to understand, the function of each genes, and then you have to test, if those genes or the enzyme are broad spectrum or flexible, so that they can take alternative substrate beyond the original substrate or the natural substrate of that enzyme. If the enzyme are flexible, presumably they can do reaction that that are not naturally done by that enzyme. All right, so it requires some effort really to do the research, and then again, sometimes the success really come from serendipity, but serendipity likes people who works hard and try, to do the experiments. I hope that answered your question.
Yes, sir.
All right.
Yes. Okay. Because of limitation of time, I think that's it for the Q&A sessions. And now, Thank you for Professor Taifo Mahmud for sharing today's material. But please stay in your position because the committee will present the certificates and we need to take the picture. Okay, for documentation committee, please proceed to take the picture. Okay, for documentation committee, please take a picture in 3, 2, 1. Okay, thank you.
Thank you very much.
Thank you very much, Prof. Taifo.
All right.
Okay, so for our next agenda, there will be sharing materials which will be given by Professor Muhammad Taher Bakhtiar from International Islamic University of Malaysia. Okay, Professor Muhammad Taher Bakhtiar is a distinguished academic and researcher at the International Islamic University Malaysia, serving as head of the Kuliah of Pharmacy with a PhD in Bioprocess from University Technology Malaysia and a Bachelor in Pharmacy from Universitas Andalas. He has held numerous leadership roles and contributed extensively to pharmaceutical education and research. His expertise spans drug delivering system, natural product chemistry, and biopharmaceuticals, reflected in his supervision of over 50 postgraduate theses and involvement in more than 100 funded research projects. He has authored numerous publications indexed in Scopus, WAS, and other database, and has received multiple awards for innovation and research excellences. Professor Bakhtiar is also a prolific conference speaker, book author, journal editor, and consultant, actively promoting interdisciplinary collaboration and advancing pharmaceutical science across academia and industry. Okay, we are honored to have Professor Muhammad Taher Bakhtiar, who will be delivering today's lecture on herbal development and delivery. Professor Muhammad Taher Bakhtiar, the floor is yours.
Assalamualaikum warahmatullahi wabarakatuh. Very good morning for everyone. Thank you, Mr. Moderator. First of all, I would like to thank University Pancasila for having us here to be a speaker in summer course in pharmacy held by University of Pancasila. So today, I would like to share with you regarding drug discovery and development to translate translational study from basic research to clinical research. Before that, I would like to introduce myself. My name is Muhammad Taher from International Islamic University Malaysia. Our base is in Kuantan. You've been in Malaysia, maybe you can recognize where actually the location of the campus. Can we share my slide, please? And also would like to welcome our colleague, friend from Philippines, Trinity University of Asia from Quezon City, Philippines. Welcome to Indonesia. Actually, I'm Indonesian, working in Malaysia. So we like to say as international local test, international speaker but local test. Okay, so our today topic is regarding herbal medicine development and delivery, regarding the translational research. As Prof. Taifu mentioned just now, it was very fundamental regarding the biosynthetic pathways of natural products, or secondary metabolites. So our university or academia, for us as pharmacy, you become pharmacy in future, our young generation to continue, our contribution to the community. So how actually to translate from basic research, to clinical use or to the community, So the role of the pharmacy here is very important. Yeah? And we are not just doing research in university to publish paper, apply some patent, yeah? But none of the community got benefit from that. Yeah? So today talk will try to discuss the process, how to do it, right? So for the student example, we don't know where to start and how to do it. Yeah? And for us as academia or lecturers in pharmacy, yeah, we got some experience, even though maybe not very effective or efficient, but at least we have something to share with young generation, yeah, especially from Southeast Asia, yeah, because we are known to be part of the mega diversity countries, yeah, including Malaysia, Indonesia, Thailand, Philippines. Yeah, we are almost at same season. I mean, always green. Yeah, and then so the question is whether our resource has been used, yeah, efficiently by the community or by people, yeah, in order to improve their health. Yeah. Of course, the answer is actually to be to be explained by us as the population in that region. So according to Fortune and Global Insight, there are very high demand, yeah, in terms of the use of herbal medicine in the market. Yeah, according to a statistic, it is about 8%. Yeah, CRGR, yeah, compound annual growth rates of the use of medicinal herbs in global. And then you see the trending, yeah, trending now is regard to the application, yeah, mainly for food and beverages, yeah, pharmaceutical and nutraceutical, as well as personal care. If you may be aware regarding the use of the traditional or natural product, yeah, in cosmetic or skin care, you can see the demand regarding the what called the active ingredient like essential oil, yeah, for fragrance, yeah, or maybe for cosmetic, you know, Centella acetica, Gatucola, or Asian pennyworth. Actually, that is very high demand nowadays regarding the skin health, especially for the cosmetic industry in Korea. You can see nowadays product from Korea regarding the skin, they add natural, yeah, natural product ingredient in there. Actually, this one is to anticipate the you know, the what called they say the side effect, the chemical to the skin, for example, they try to replace, yeah, including for pharmaceutical as well, as we experienced in COVID-19, yeah, in 2020, yeah, it was started in 2019, and also regarding the supply of the drugs, yeah, or modern medicine at the time was very affected, and also people try to find alternative. I don't know, they explained by the friend from Philippines, but your people also trying to find something from nature, right? To overcome the problem at the time. So you see the trending now. The main player here is a country in Europe. You can see the amount of money that actually contribute to the can say as trading, also manufacturing, is about 103 million US, yeah, in 2024. And then what is actually what is the effect to us, yeah, us as developing countries, the mega source, can say mega source of the botanical and natural products, high demand, yeah? High demand of the active ingredient from developing country like from the Southeast Asia, China, India, yeah, and maybe Brazil, yeah, the biggest diversity in the world is very high nowadays. You can see it's not just what called Yeah. It is actually a fact that you can see the Europe. Europe is the biggest player in the use of herbal medicine. Yeah. You can see the projection. Yeah, projection is very promising. Yeah, promising here. Even though the market is Europe. Yeah, but we as in our region, yeah, we can supply many things. Yeah, like essential oil, for example. You know, gaharu in Indonesia we call it gaharu as a component of the fragrance and perfumery. Yeah, and also maybe Pegasus now, Centella acetica, yeah, we can supply. Then, so why we, I can say, left behind? Don't be mad to me. Left behind means we are not progressing like them. Yeah, like them here is European or maybe China or Japan, for example. Here is actually related to the political will, yeah? Policy maker, yeah, because it is related to what call the things that need to be regulated. So why we are not able to live, yeah, to live up, yeah, the herbal product to be modern medicine, for example, or to as a replacer, can say a replacer to the modern medicine because of political will as well. Because when we are committed, yeah, our commitment to develop must be come out with regulation. How we can monitor, yeah, how we can control the product in the market, because it is will be what called it will open for many misuse, misapplication of certain active ingredient to the natural product. So who involved in translational? Yeah, translational means we would like to translate finding from basic into clinical. Yeah. So who should involve is medical practitioner. Yeah. We have product sample. If you don't want to use it, our product will not go anywhere. Yeah. And then of course policy should support. How policy regulation, the government can support, can protect the adulteration, for example, and then research. Yeah, research is very important. Research is here. Yeah, research is for us. Yeah, we are strong at research. Yeah, the role of the academia, researcher in university will be unlimited actually. Yeah, because we have a student, we have a doctor, we have associate professor, we have professor. Means we are having actually advanced knowledge, yeah, regarding how to develop, how to prove, yeah, how to provide things that actually required, yeah, in order to develop one product. Then one big question actually, yeah, if we are in the process of drug development, yeah, the big question whether the first question whether want to have disease first or drug first, yeah. So any answer for that, yeah, drug first or disease first? Either, yeah, yeah. So what do you think? Yeah. So meaning here actually can be the question can be, you know, there's nothing, no correct, no wrong, right? Yeah, answer, yeah, because you can see like what happened in COVID-19. COVID-19 actually taught us very what call it the good experience regarding how to handle this situation. Yeah? So in this case, COVID-19 meaning disease first, right? Disease first, and then we come up with vaccine, yeah, and also maybe we can still utilize any antiviral drug, yeah, in the development process. Then the things that I would like to highlight here is regarding why there is a big gap, yeah, big gap many research. Yeah, we did many research, many publication, but not many product. Yeah, not many drug available in the market. What happened? Yeah, what we call it in this matter is we call it valley of death or gap in translation. Yeah. So from academia meant mainly for basic research, and we want to convert it into industry. The industry meaning this already in clinical setting. Yeah, because industry play important role in the development of drug at phase 1, 2, 3, and 4, isn't it? Because it requires amount of money, yeah, to develop. Yeah. So the main culprit, yeah, what to me, we call it culprit, yeah, something that can be say their mistake. Yeah. Example, there are some, yeah, some or list of the culprit can be say here. There is reproducibility. Yeah, we are in academia doing research. Sometimes our method that we use maybe not robust method. So when we try to reproduce, to repeat the same, we are not successful. Yeah. So this one also one of the reason, yeah, for valley of death. Yeah, regarding the drug development. And also clinical relevant. Well, clinical relevant means whether it is really needed or not. Yeah, structural and many other reason, yeah, of this gap. Then if we see, yeah, based on the history, actually there are many medicinal plant heritages, yeah, or wisdom, yeah. So the most what you call the popular one, you got Kempo in Japan, yeah, TCM in China. You can see how TCM nowadays can actually, I say, conquer the market. I'm not sure whether it is in Philippines also, yeah, but in Malaysia, Indonesia, the traditional medicine are mainly from TCM, Chinese traditional medicine. Yeah, Ayurvedic product, but not is available, but not much here. Yeah, and also traditional Arabic and Islamic medicine, and we have traditional wisdom here. Brother, maybe sister, also lecturers. We call it jamu. Yeah, jamu is Indonesian traditional medicine. Yeah, it was actually been used for long ago. Yeah, but they are not very good in progress. Yeah, they are not go anywhere because, as I mentioned just now, some restriction. Yeah, to develop it. Perhaps regulation also play important role here. Yeah, to slow down the process. Yeah. Then we see regarding drug and herbal discovery, yeah, or development, it almost like drug development as well. But perhaps differences maybe in terms of the approval process, maybe less about little bit lenient, isn't it? If compared to drug, yeah. But it still needs to undergo some process like discovery process, preclinical as well, yeah, clinical, and also even to sell the product to the global, you have to undergo FDA review, yeah, and also we need to check whether it is, they don't have any side effect, yeah, in future. Then if you see the main challenges, yeah, main challenges in the developing, yeah, traditional medicine, especially jamu in Nusantara, yeah, in our region, you call it Nusantara. I don't know whether you are familiar with Nusantara. Nusantara is our region. Southeast Asia region, you call it Nusantara. Yeah, we are part of Philippines, yeah, a part of Thailand, yeah. Malaysia, Indonesia, we call it Nusantara. Yeah. So because they share the same knowledge, yeah, same wisdom, same skill, yeah, we call it Nusantara. So the challenges mainly dosage, yeah, doses, inconsistent doses, because we don't really what call control, yeah, regarding we don't really record sample. Yeah, we do not like traditional Chinese medicine that already it's call it a good record. So doses sometimes different, and then they don't have the same activity compare from one people and other people. Stability as well, stability, because we are dealing with extract, maybe this containing water, as pharmacy, right? Water can affect the stability of the active ingredient. And then standardization, what compound should be there, how much to be there, for example, because when we are talking about the extract, we have to know what compound to be monitored, how much, at least, the compound to be in the product. It is not not run away from the drug receptor interaction. For herbal, they are also working like drug. But drugs need to meet receptor. The amount of drug to meet receptor should be sufficient enough to give some clinical or pharmacological effect. So sometimes we don't rely on don't really care about the standard. Of course, they are not giving any effect. And then to hygiene, the preparation. If you know jamu, jamu in Indonesia, they are freshly prepared. In general, jamu, they are freshly prepared. They combine some rhizome, or maybe leaves, you prepare, and you can use it directly normally in the morning. If you have time, we have visit, right? Visit to Tawang Mangu, we can actually see the people who are practicing, using traditional medicine, call it as jamu, especially in Central Java. And then next challenges is regarding additive, adulteration. To make sure there is no additive, we sell the product, for example, this one is for anti painkiller, for example, or anti-inflammatory. Make sure there is no steroid there, make sure there is no drugs in the product. This needs to be controlled. Then regarding the commercialized traditional medicine, especially in Indonesia, they can you can find it in form of extract, direct extract, we call it jamu, and then you can find it in terms of standardized active compound, we call it obat standar, and then you can find it in, this actually level of traditional medicine in Indonesia, raw extract, we call it jamu, or next level is standardized, we have identified what is active component there, and then measured dose, call it biopharmaca, that need to be produced through GMP facility process, and then the stability has been increased perhaps by doing some kind of formulation sample. Then herbal medicine preparation can be found in form of extract, decoction, tincture, syrup, and also eye wash. I already briefed you regarding the trending in global market, but here is additional information regarding the importance of traditional medicine. You see, in Africa, 90% people, and India, 70% still depend on traditional medicine. And then if you see the history of the traditional Chinese medicine has been used 3,000 years ago. Then top selling botanicals, is actually with G, ginkgo, garlic, and ginseng. 3G actually, they are we call it the 3G, ginkgo, garlic, and ginseng. And then you see the 127 drugs approved worldwide, for treatment cancer. 70% of them are based on natural product. You can name it, like vincristine, vinblastine. What else? Doxo, also from natural product. One is from microorganism. So meaning, what we can highlight here, because the importance of natural product to be preserved and to be developed. Yeah. And then next, what you call it, regulation. Every country they have their own regulation. How they can utilize their own resource to be used in the healthcare. And you see in Indonesia, they need the diversity. 30,000 plants potentials can be used. Still being used by 70% of people in rural area. And then kind of disease that can be treated. Can be cancer, arthritis, cholesterol, stroke, diabetes, and so kidney disease. Talking about traditional medicine, sometimes there is pro and con, isn't it? Many pro and con. They said traditional medicine is or cannot replace. But perhaps the things, the wrong things here, because we don't really control regarding the dose, regarding the standardization of the herbal medicine that can be used to treat certain disease. This actually regarding the fact that small molecule from nature that can be used for. You study about the mainly the anti-cancer are derived from natural product, isn't it? Like let's say from Taxus brifolia, they produce paclitaxel, and vincristine, from Vinca rosea. So meaning role of natural products, role of herbal medicine is very important in the process of drug development. Then what is the global challenges in drug or herbal discovery and development nowadays? What we need to find nowadays is related to anti-cancer, targeted. We have combine of anti-cancer with different mechanism, but whether they are targeted or not, they are targeted means they are going directly to the site or not, mainly they are going systemically, to the whole body, so they are not very effective and efficient to kill the target, the cancer target. So meaning targeted means we have to work in delivery, how we can deliver the drug directly to the target. And so here, what kind of innovation maybe we can to improve the delivery of the anti-cancer drug. Example, we can make them in the what do you call it, the conjugation, like antibody drug conjugation, to bind drug with antibody. Because antibody is very specific to recognize antigen. Surrounding the cells, cancer, they have a certain antigen that can be recognized by antibody. So the antibody can carry the anti-cancer drug to the target. So the drug will be released at the site, so they can be working locally to the target. Then antibiotics also one of the challenges nowadays. As we know that antibiotic is derived from natural product as well. From biotics. Biotics meaning from fungus. So the challenge now is because many antibiotics nowadays is really not really effective to certain microorganism. So we have to try to find another generation to generation in order to overcome this problem. Then vaccine. Vaccine also becoming one of the, even though we are experiencing COVID-19, but we don't know what to come. Have you heard about the Disease Act? Disease Act, something that unknown disease and now unknown pandemic. The trending, meaning we have to be ready with this. Yeah? Even we have experience in COVID-19, we have some kind of procedure, yeah, to handle it, but we have to ready go on a next, yeah, pandemic if it is happen. Yeah. Then next is to improve the recovery. Yes, now we have actually you just name any kind of disease. We have drug for this, right? You name it. What kind of a disease, for example? We have already drug for that. But actually, whether the question is whether the drug is effective and efficient, yeah. Whether they are having low side effect, yeah. So our task is actually to increase the efficacy and to reduce the toxicity of the current drug. So never stop, yeah, never stop, yeah. As pharmacists, we should never stop in doing innovation, yeah, in doing research, yeah, in order to improve the efficacy and efficiency, yeah, of a drug. Then Prof. Taifu already mentioned just now regarding the source of drug. It can be from natural products. Yeah? Prof. Taifu is very experienced in doing a kind of modification in the biosynthesis, yeah, of natural product from plants, yeah, from chemical synthesis, yeah, biosynthetic pathways also through biotransformation and through recombinant DNA technology. So nowadays recombinant DNA technology is becoming one of the important. Yeah, if you see the top selling pharmaceutical product nowadays, yeah, is we call it Humira, Humira, or maybe fact is actually that one is monoclonal antibodies. Yeah, they are actually made from recombinant DNA technology process. Yeah? Okay, and you see the small molecules, yeah, and macromolecules. Yeah, what's differences between small molecules and macromolecules? Yeah, let's say we have kind of one disease like called diabetes. Yeah, how to manage diabetes? Normally we use drug. Yeah, actually to control blood glucose level. Yeah, the alternative treatment is using small molecule or macromolecules. Yes, actually macromolecule is insulin is to be used for patient with diabetes type 1, isn't it? Actually insulin dependent, yeah, insulin dependent. But it is replaceable at certain condition, yeah, with small molecules, for example metformin. Yeah? Just to give you some idea, yeah, because some idea with you for those who are not having any exposure to this. Yeah? So alternative can be macro and small molecules, yeah? Especially for diabetes. You can see some differences between macromolecules and also small molecules. So regarding general procedure to develop new drug from plant source, yeah, I think this one is actually the common chart, yeah, common chart. Where to start? Yeah, of course we have to start from material, front material first. Yeah, we do extraction. Yeah, and then we do bioactivity testing. Yeah, structural characterization, as well as what next is actually clinical study for the active component. Let's see about the in vitro study first. Yeah, in order to do kind of translational research, yeah, from basic research to clinical research, yeah, in university, yeah, in academia, we do a kind of what called bioactivity study. Yeah, bioactivity study can be done through in vitro, yeah, in vitro and also in vivo. Yeah, in vitro study, basic research, sample using chemicals, yeah, chemical or reagent or enzymes, yeah, or we can use microbes and also cell lines. Yeah, so from this experiment, what we can actually obtain, yeah, we can prove some activities, yeah, basic basic data regarding the activity of the extract or active compound. Example for antioxidant, anti-diabetes, yeah, anti-inflammatory, yeah, anti-cancer, and so on. Yeah. So in vitro study is actually the preliminary, yeah, before moving forward, yeah, to in vivo and also clinical study. We have to have in vitro study. Yeah. This is really my experience regarding the study of the use of cinnamon. You know cinnamon? Yeah, cinnamon selenacum. Yeah. This really very good for traditionally, yeah, very good for diabetes. It can lower blood glucose level. Yeah. So for us in academia, what should be done? Yeah, because of traditional wisdom, for example, yeah, people told you that this plant is good for cancer. Yeah, this plant is good for antibiotic. What should be done? Yeah, we in university or academia, we should have kind of systematic process. Yeah, of course we need to do kind of extraction, isn't it? Because we cannot just use raw material, yeah, to be tested, yeah, especially for in vitro or in vivo. Yeah, we need to extract to find a compound, yeah, test them in vitro and in vivo, and after that we can move to clinical study. Yeah. And also we can do kind of what called wound healing study, yeah, using cell line. Yeah, you can do scratch assay, yeah, to check whether the plant extract or active ingredient can be used for wound healing. Then animal study. Yeah? So in animal study is actually the continuation of in vitro study. When you prove that the called the extract sample or active ingredient is good, yeah, in vitro, so we need to prove it at in vivo level. Yeah? You can use different type of animals, yeah, like monkey or maybe rats, yeah, mouse, yeah. Also can use bird even. Yeah? Then we have to follow the guidelines. Yeah? There are some guidelines that we follow, yeah, when we use animals, yeah, in vivo study. Yeah? OECD guideline, NIH guideline, yeah, ICH, CPC, and next is actually we have to find to the rules. Yeah, there's a rule in vivo study. Yeah, what's called it reduction. We cannot just simply use number. Yeah, choose any number when we are doing in vivo study. We have to reduce. Yeah, that's why the role of the ethical committee at university level actually to monitor the use of animal, yeah, in clinical study and define, yeah, whether it is really need or not, and also replacement. Yeah, that there's some sort of question. Yeah, when we try to use animal in our study, there's some sort of question need to be answered. Yeah. Then what effect can be obtained from in vivo study? Yeah, can get pharmacokinetics profile, pharmacodynamics, physiology, and also pharmacological effect. Yeah, can be used for many type of testing. Yes, yeah? Like anti-hyperglycemic, yeah, wound healing. Yeah, and the goal of this is to understand, yeah, the mechanism of action of the active ingredient or extract. So herbal extract or active ingredient derived from natural product. Actually, if you see biopharmaceutical classification system of them, BCS classification, still remember BCS class, yeah? They are 1, 2, 3, 4, yeah? Mainly they are at level, they are class 2, low solubility and high permeability. Most of them at that class, yeah? So their problem is regarding their solubility, yeah? Water solubility, but they are good at what called permeability. Yeah. So meaning we have to do some kind of innovation. Yeah. Actually, to improve their solubility as well. You can use many type of what call it innovation nowadays. Yeah, we call it nano formulation. Whether you want to use this liposome technology, yeah, and many other type of technology actually to improve the delivery. Yeah. So this is really very important as well, yeah, in the development process, yeah, because the failure in drug development mainly because of we are not using the good formulation or delivery process in order to carry the drug to the target. Yeah, so meaning some kind of innovation and formulation is important. Yeah? For example, nisomal formulation. Yeah? Nisom is using called kind of it is like liposome, yeah, losing physicals. Yeah, nisom is good because it can pass through skin barrier. Yeah, it can pass through skin barrier. Let's say your product is good for skin, but we are not deliver it in appropriate formulation. Of course, they will not show any bioactivity, yeah, to the to us, yeah, to us. Okay. Then one need to be monitored also in herbal development is legislation. Yeah, I mean regulation, how to support. Yeah, because as mentioned by vice rector just now, yeah, the word natural, yeah, the promotion regarding the natural is good. Yeah, natural, everything related to natural is good. Yeah, but sometimes it is actually conception. Yeah, conception sometimes not all natural is good. Yeah? Then based on the data, yeah, 123 million, yeah, people still use product for various purpose, yeah, like obesity, to prevent cancer, yeah, to ease pain and so on. Yeah. So meaning herbal regulation is play important roles in order to improve, yeah, development of natural products. There are three fundamental principles, yeah, in herbal development. The first is standardization and regulation, yeah? And then scientific proof of beneficial clinical effects and scientific proof of safety, yeah, and acceptable toxicity, yeah, for the patient. Yeah? So we have to work on this, yeah, in order to give some evidence, yeah, for the development of herbal medicine. And then regarding the quality, yeah, of the products, yeah, sometimes it is difficult to make sure, yeah, dose, yeah, and also standardization of the active ingredient because it is affected by method of extraction, yeah, method of whether it is using water or using chocolate, yeah, or maybe maceration, for example, yeah. Then time of extraction, yeah, temperature of extraction, yeah, solvent itself and solvent concentration as well, and polarity. Yeah? So it will affect the quality, yeah, and quantity of certain active ingredient in the product. This is really for general. Yeah? There are some methods of extraction that can be used. Yeah? There are many options. Yeah? Depend on a target compound. So when we try to extract certain active ingredient, yeah, from certain plant sample, yeah, we have to understand the nature of the active ingredient, yeah, whether they are heat labile or not, yeah, whether they are soluble in certain solvent or not, yeah, we have to check, yeah, so we can decide, yeah, what method to be used in the extraction. Next, in order to make sure, yeah, the product is actually well protected, yeah, so I have to do quality assurance and also quality control, yeah, regarding the identity, yeah, identity of the products, yeah, through pharmacognostical testing. I think you learn in pharmacognosy, yeah, how to make sure, yeah, how to identify, yeah, the product is the correct product sample, yeah, especially for raw material, yeah. Can check through microscopic, yeah, and and also can be used qualitative assay, yeah, and purity of compound, yeah, purity of compound can be checked using SPLC, yeah, content instrumental using SPLC, yeah, microorganism content, yeah, can be checked through microbial growth test, yeah. Stability testing, we have to follow, yeah, ICH guidelines. So regarding variable composition, yeah, of the herbal medicine or herbal plants, yeah, herbal product, yeah, so herbs do not have consistent and standard composition. Yeah, it is depend on the geographical, yeah, geographical farming sample, yeah, and also there are numerous chemical consistent in different part, yeah. So that's why we have to select where is the, you know, the highest, yeah, highest content of the ingredient, whether it is in the stem, leaf, fruit, yeah, or roots, yeah. And factors can affect the availabilities can be climate, yeah, growing condition, and time of harvesting. So need to be properly monitored, yeah, need to be properly monitored and properly recorded, yeah, regarding this factor, yeah, climate, yeah, growing condition, time of harvesting, and also for harvesting as well as storage. Yeah? And next, we need to check the contamination. Yeah, contamination can be from because of misidentification of species. Yeah, misidentification of species. And sometimes we have same local name, yeah, but maybe different species name. Yeah, need to be checked properly. Yeah, and then adulteration, yeah, addition of maybe any drugs to the product, yeah, and heavy metal and pesticide content, yeah. So marker compound need to be checked, yeah. If you are familiar with Andrographis, yeah, Andrographis paniculata, yeah. Sample, we have to make sure what is the activity to be checked, yeah. Andrographis paniculata is actually quite familiar plant, yeah, in traditional medicine in Indonesia or also Malaysia, yeah. Need to be checked like andrographilate, yeah. The compound is andrographilate, yeah. So we have to check how much andrographilate should be there, yeah, yeah. How much andrographilate should be there, yeah, and then we need to monitor time to time, yeah, in order to make consistent, yeah, consistent ingredient in the product. Then analytical inequality control, like using sophisticated equipment, yeah, it is important to monitor, yeah, to make sure the quality of the product using SPLC, gas chromatography, yeah, and also LCM as fingerprint. This just additional, yeah, regarding the actually nowadays we call it metabolomic, yeah, metabolomic study, very specific to certain plants, yeah, like fingerprinting, yeah, like can be used for fingerprinting for certain product, so we can use this process, yeah, through this flow chart. Okay? Then this fingerprinting, yeah, meaning when we run SPLC LCMS to stain extract, we have to have this kind of profile, yeah. Not necessarily to have same amount, but we need to have same profile, yeah, same profile between one batch to other batch, yeah, in order to make consistent consistent ingredient in the product. So now for conclusion, yeah, for this today talk is WHO still promotes the use of traditional medicine, yeah, particularly herbal medicine in primary healthcare. Yeah. And then some countries still rely on herbal medicine in their primary healthcare, yeah, and herbal medicine development requires scientific. Yeah? So this is actually our role, yeah, to provide scientific evidence, yeah, to support the development of herbal medicine to move forward. Yeah, can be used to replace modern medicine or to be used to give some kind of supplement, yeah, to modern medicine so that it can increase um, uh, human well-being, yeah, to improve quality of life of the people. Yeah? So I think that's all from my side. So thank you very much. I will welcome if you have any question, query on this topic. Thank you very much. Yeah, please. Yes, please. Yes.
Penetrate because I'm really interested in dermatology. So when you mentioned nisomal formulations and penetrating and helping topical applications or transdermal applications penetrate, right? I'd like to ask, how are these systems typically developed? Because I'm really interested to integrate nisomal formulations um, in drugs.
Okay. Thank you very much for your question. Very good question. Nasal formulation is mainly being used for skin health, because you know that skin is important organ in our body. They protect us very well from any environmental factor, out you know, through mechanical, yeah, also chemical. So we should thank God to have skin, isn't it? You can imagine you don't have skin. What happened? Okay, maybe we just like sponges, yeah, can grow, swelling, can be, you know, smaller at the time. But that's why our in pharmacy, we are working mainly to, can say, to strategize or manipulate. Yeah, the alternative how to deliver drug to the skin is using niazom. Yeah, niazom is actually physical substance containing surfactant, yeah, and also phospholipid, yeah, because you know surfactant is actually ingredient that can reduce surface tension. Yeah, they are good. Uh, they are good to, you know, to interact with fat. Yeah, they're good to interact with fat. So the drug can be formulated in that vesicle. It is like round, yeah, round vesicle. The drug is trapped inside. So because of the nature of the surfactant, they can pass through the skin. Yeah. So they can pass through the skin, meaning we can actually bring the drug to the target. Because you know the skin layer, isn't it? We have few layers of skin. So you target where actually the target, whether it is to treat the surface, middle layer of the skin, or maybe a deeper layer of the skin. So niazomal is actually quite popular nowadays. Cosmetic, yeah, cosmetic. Let's say product from Korea. Yeah, I don't know whether product from Korea also available in huge number in Philippines. Yeah, but in Indonesia and Malaysia, product Korea mainly they try to formulate it in niazomal formulation in order to deliver the active ingredient. Yes. Is it? Yeah. Okay. Any more question, or do you still have time, or just already? Thank you.
Okay, thank you so much, Professor Muhammad Taher. Because of limitation of time, I think that is for the Q&A session. Okay, once again, thank you very much, Professor Muhammad Taher Bakhtiar, for sharing today's material. Kok mau kayak gitu?
For our third material will be given by Associate Professor Denny Susanti from International Islamic University, Malaysia. Professor Denny Susanti is an accomplished associate professor at Kuliah of Science, International Islamic University, Malaysia, with a distinguished academic background in chemistry and pharmacy, including PhD from University Technology Malaysia. Her extensive career span teaching, research, curriculum development, and administrative leadership, including roles as Deputy Dean and Head of Department. She has supervised numerous postgraduate theses, contributed to over 50 funded research projects, and authored a prolific body of scholarly work indexed in Scopus, YOS, and other databases. Her research focuses on natural products, drug delivery system, antibacterial agents, and wound healing, with notable contribution to cosmetic science and medical chemistry. Professor Denny has also served as a journal referee, conference organizer, and advisor to academic and governmental bodies, earning multiple awards for her educational and research excellence. Now, we are honored to have Associate Professor Denny Susanti, who will be delivering today's lecture on integration of artificial intelligence, or AI, in natural product research. We would like to remind for the presenter, the time will be given is 35 minutes for the material and 10 minutes for the question and answer session. For Professor Denny Susanti, the floor is yours.
Okay, Bismillahirrahmanirrahim. Good morning to all the participants, from Philippines and Indonesia. I think you are tired now. Yeah, can you stand up all before I start my presentation? Can you stand up? So we'll make refresh ourselves. Yeah. So just a student. Yeah. So just for student. Okay. Okay. So can you choose your partner? Choose your partner. Yeah, choose your partner. Okay. So hold his or her shoulder. And then try to massage. Okay. And then take turn, for that. So you feel relaxed after the series of lecture. Okay. So how you feel now? So you can move around to make your more relaxed. Yeah, because I think 1 hour lecture is quite boring. Yeah. So try you move around before we start again. Okay. So if you feel relaxed, so you can sit down and we start again. Okay, thank you. Okay. Okay, thank you for the master of ceremony, for the nice introduction. Yeah, during my I don't know if I say this a lecture or just, I think this is just a sharing, So you can eat, You can eat, you can drink, So usually in my lecture, in my university, I allow my students to eat, because sometimes, our class is 8 a.m. They don't have time to get their breakfast, so I allow, So I just to take water or a bread, during my lecture. Okay, so I allow you to do that. Okay, so okay, can you share my slide? Okay. Okay, so I don't know why I choose this topic. Yeah, so but this topic is quite hot now. Yeah, using AI. Yeah, I think some of you, you use AI for maybe your assignment. Yeah, or to draft your assignment, or just to ask. Yeah, to ask a simple question. What is it? What do you think about this term? Yeah, so we use AI. Yeah, so AI become our habit to us. Yeah, so before this AI came, yeah, we have a Google, right? So everything we will Google. Yeah, we will Google to us everything, and then there is a new technology in computational. Yeah, so I think they make some apps or system, yeah, to help us. Yeah, not only for the simple tasks. Yeah, so AI also can provide, yeah, the system or apps that will help us during our maybe research, yeah, or especially, yeah, for you as a student, if you have a final year project, yeah, so you need to do the task, yeah, to do the research. So AI actually can help us, yeah, for maybe to optimize our result. Okay, so for AI, yeah, when we see there is a development of computational omics. You know what is omics? Do you know what is omics? Okay, so omics is the field in the biological study. Yeah, so biological study where there is a material that present in the living organism. For example, protein. Right? What else? Gene, metabolites. So this produced by the living organism. Yeah, living organism here. So meaning that not only microbe or plant, but also human. Yeah. So if the biological study is we are focused on gene, we call as genomic, right? So if the biological study that we study is a protein, we call as proteomic. Yeah, so relate to the natural product. Yeah, so we focus on metabolites. Yeah, secondary metabolites. Yeah, so the omics we call as metabolomics. Yeah, so the field of metabolomics. Yeah, this quite good progress now. Yeah, because of the development of the computational. Computational. Yeah, so from the computational. Yeah, so we can get the prediction. Yeah, the prediction here, the prediction in terms of the reaction. Yeah, and so on. Okay, so and then after that, yeah, so first is computational. Yeah, computational science. Yeah, after that there is the AI, artificial intelligence. Yeah, so artificial intelligence. Yeah, so they are copy like our intelligence, the human intelligence. Yeah, so how our intelligence works. So this how artificial intelligence works. Yeah. Okay. So so this we can say as the computer system. Okay. So and then this AI we can integrate. Yeah. In the natural product research. Okay. Okay. So next slide. Okay. So this morning, yeah, Prof. Taifu have mentioned about the source of the drug, yeah, and also the how long the process of the natural product research, yeah. So even Prof. Tahe just now also mentioned about the process for each process until we discover the active drug. Yeah. So you know how long process start from the extraction until we got the single compound, and then I just mentioned here, we start from bench in our lab until the bedside. Bedside meaning the drug that we are ready to take. How long the process? How many years the process? You know, how many years? Can reach 20 and 25 years. So you see, yeah. So if you start now, how old are you during that time? 50? No, right? 40 maybe, or I think 40s. Yeah. So this actually how long the process. Yeah. So this one, when we use conventional method. Yeah. When we use conventional methods. Yeah. Start from we have a preliminary screening from plants, yeah, or maybe this plant used for as a traditional medicine in our country, yeah. So we test, we do the screening, and then we see what is the activity or what is the compound. To know the compound, we need to extract, we need to isolate, right? And then we need to do the bioactivity, and then to see if this traditional medicine, we need to confirm. For example, this plant is used. This plant is used for to treat head, like analgesic, for example. Yeah, analgesic. So to prove that, we need to do the bioactivity. Okay, so after that, after we got, we know we got the single compound, we know the and then confirm the bioactivity, and then we need to know, what are the structure? What is the structure of compound? Yeah, so do you know how to elucidate the structure of the single compound that we isolate? What is the method that we use to we need to use? There is IR, right? There is infrared, there is ultraviolet, there is NMR, MS, until we got the structure. Yeah. After that, there is a serious process that have been described by Prof. Tahir just now. Process to produce this drug, this compound to a drug. Yeah, so you can imagine how long, the process of the from bench, from a bench of our lab until the bedside. Yeah, so with AI we can shorten the process. Yeah, with AI we can shorten the process. Okay, so as we know that this natural product, this become a central rule of drug discovery. Yeah, so this one have been explained by Prof. Taifu and then Prof. Tahir just now. Okay, so even around the 60% of the drug that available now, so maybe come from natural product, or maybe come from its derivative, for example, like Fakultas cell just now, this actually derivative. Yeah, so or maybe the idea, the idea of structure inspired from the natural scaffold. Yeah, so this is the drug that available now. Okay, so like I mentioned, there is a long process. Yeah, there is a long process to get the drug, after we after we produce from the natural product. Okay, so next slide please. Okay, so this is the history. Oh yeah, so this is the history of the. So this is the history of AI, in natural product research. Yeah. So they start with the computational computational. If you, I don't know if you are aware that there is a subject about computational chemistry or computational aided drug design. Yeah, so this the process, the first started of AI. Yeah, so there is a software or system. Yeah, so computer aided drug design. So where this system we use as a part of drug design. Yeah, so we get the data from the nature. Yeah, for example, so maybe we isolate of the compound from nature, but maybe the physical chemical properties of this compound is not support us to design the drug. So maybe we change or we modify the structure. So with the help from the CADD, so we can design to improve the physical chemical properties of drug, but still have the bioactivity that we need. Okay, so this the first of the using of the, we can say the computational chemistry. Okay, so and then the recent advancement for the using of AI, okay, so this increase the capability of CADD. Capability of CADD. They're not only for CADD we just modify, maybe modify the functional group, sums, and then we see the activity. So this for CADD only, So but for AI, so this increase the CADD capability more deeper. So I will show you later on. So and then the increase the capability of CADD. But actually this not just based on the human perception. So kalau if we use human perception can be wrong. But if we use this AI, so this we can escape from the human perception. Okay. So what we see now, so before the AI come across, the research on natural product, so there is a lack, in terms of the exploration, okay. So because there is still, the AI is still in the beginning, So sometimes, we isolate, maybe from a plant or from microbe, so, or from ocean creature, or from animal, So we have a lot of chemical compound, So but sometimes after conducting several research, we are stuck. One, because of there is the compound that we isolate, there is no bioactivity. And then the second one, the compound that I isolate active but very toxic. And then compound that we isolate, the third one, the compound that we isolate, active but have a poor physicochemical properties. If poor physicochemical properties, it's difficult for us, to study on the ADME, absorption, metabolism, excretion, distribution, and excretion. So this is difficult. Okay, so with this, during that time, the research on natural product, they're stuck. They're stuck. Yeah. So there is a time. Yeah. There is a time. Yeah. When I on this field. Yeah. There is a time. I just isolate, elucidate the structure, and then do the bioactivity and write in journal and publish. Yeah. So this just what we done because we don't have any tools that help us to do beyond that. Okay. So and then when the artificial intelligence come, we can do anything. Yeah. So this one. Yeah. So if you see here, so this the artificial intelligence. Yeah. So artificial intelligence. Yeah. So this the what we call as all the system, the broad. Yeah. So they can we can ask anything. Yeah. They can give us answer correctly. Yeah. We can do the artificial intelligence can do the prediction. Yeah. Prediction. Yeah. So they can do anything. Yeah. For the artificial intelligence. So this is the biggest picture of AI. Yeah. And then sub of the AI there is a machine learning. Yeah. Sub of the AI there is a machine learning. Okay. So the machine learning. So this a part of the subfield of AI. Yeah. Subfield of AI. Okay. So from the machine learning. Yeah. So this focus on algorithm. They focus on algorithm. Okay. So when they are focused on algorithm to the data that we supply to the machine learning. Yeah. So this machine learning. Yeah. Can do the intervention. Yeah. And then after that, they can give a prediction. But for machine learning. Yeah, sometimes need intervention from human. Yeah, because sometimes they predict in the wrong way. So the machine learning still need the intervention from human. Yeah, for example, we do all the we give the data, but suddenly this not like what we need. Yeah, what we expect. Yeah, so we need to change. To do, meaning that we need to do all the experiment again and then input the data to the machine learning. So this can come up with the our prediction. Okay, so machine learning need human intervention. Okay, so the subset from the machine learning, so there is a what we call as a deep learning. Yeah, so deep learning. So for deep learning here, okay, there is no human intervention. Yeah. So for one of the example for the deep learning. Yeah. So this one they copy like our neuron. How our neuron work? How our neuron work? When our neuron got the signal. What happened to the signal? The signal will be processed, and then after that will be transformed, right? To transform to the response of any muscle. For example, the signal that you receive, will make you cry. So there is a response in our neuron. So after that, this neuron will send a signal so to make our tears come up. Yeah? So this how the neuron work. So this similar to the deep learning. Yeah. So we input the data to the deep learning. Yeah. So the deep learning will digest. Yeah, will digest and then will see everything. They will see the effect, what effect of this input, and then will produce output. Yeah? So this actually the deep learning. So I will explain later. Yeah, more detail. Okay. So the next slide. Yeah, I will make sure this this presentation. Yeah. So simple, fast, and sweet. Okay. Okay. Next slide, please. Okay. So this the application that I can show to you. Yeah, for AI. Yeah, in the nature product research. Yeah. So first, yeah, AI is used. AI is used to mine, for mining. What is the purpose of mining? Collect everything, right? Yeah. So AI will mine all the info. Yeah, in terms of genomic, in terms of metabolomic, proteomic, all the omics. Yeah, all the that produced by the chemical compound that produced by the living organism. So this become a mining. Yeah, become a mining. Okay, so and then yeah, after this not only the omics, yeah, but also maybe yeah, in the Prof Taifu lecture just now this morning, Prof Taifu present on the biosynthetic process of the natural compound. Yeah, so for AI, yeah, so we also mine. Do the mining, yeah, for the all enzyme that responsible for the production of the chemical compound in the living organism. Yeah, so you know what is the what is the natural what is the antibiotic actually produced from what? Antibiotic from what? Antibiotic produced from microbe, from fungus or fungi. Okay, so this the antibiotic. So there is an enzyme that responsible to produce the antibiotic. So to understand I mean the good thing if we want to work on that AI, we need to understand the biosynthesis pathway for the all the chemical. Yeah, so we know what is the enzyme that are responsible. Yeah, so here AI also can use to mine all the enzyme that are responsible. Yeah, so we have all the genome and metabolome mining. Yeah, so all these things. Yeah, so I will show it to you how to connect this one. Okay. Okay, the next slide, please. Okay. So if you see here, yeah. Okay. So if you see here, yeah. Okay, so these all the mining, yeah. So there is a chemical compound, there is a enzyme, yeah, that responsible. There is this what we call as genome, proteome, metabolome, anything omics, yeah. So this one. So this our mining. Okay. So from this one, actually, we can use, so we can use, all this data. So this all data. So you can see here, AI is more on data. So how we get the data? For example, like a metabolomic, we got the data. So we got the IR data. We got the NMR. We have MS, we have UV. So this all data. Yeah. So for one compound. Yeah, for one compound. Okay, so we have all the data. So we have the MS spectra, NMR spectra, UV, IR. So this become a data for AI. Okay. So and then gene. Yeah, gene here that used to produce enzyme for the biosynthesis of the compound. Yeah, so we have this all. In actually, what is the action of the enzyme? Is it oxidation, or reduction, or hydroxylation, whatever? for the how the enzyme work. So we have the data. Okay, so and then we can use this one. This one is we can say quite conventional artificial intelligence. So for this we can say as a part of the machine learning. To connect this all data with the biggest molecule. Meaning that the interaction with the small molecule with the bigger molecule to predict the bioactivity. So we have conventional machine learning, we have machine learning, and we have deep learning. So we will get we see the connection between the small molecule that maybe we isolate, and then with the biggest molecule. Maybe the big molecule here, for example, how this enzyme, how this this small compound can interact with hormone or enzyme, specific enzyme, to give the action to our body. So this the task of the machine learning and deep learning. Next slide, please. So here, so I just give the example. So this compound, this all compound, actually this produced from AI. This, for example, the small compound, the small compound, So this small compound, So actually this produce the researcher, they try to see the activity of this compound, the small compound, and then try to see the connection with the gene. What is the gene or what is the enzyme that responsible to synthesize this compound? So and then after the research that conducted, they see that like the original compound is not have a good pharmacokinetic properties, and then they try to modify. And then by helping of the enzyme. Still helping by the enzyme, in the system, So and then they produce this compound, and then you can synthesize it. Even this not produced in nature, but using AI, so we can this compound after the modification, this compound have a good pharmacokinetic, so we can synthesize, and then this can be a drug candidate. So and then so this the small, and then this bigger compound, this also come from this process. They not produce naturally, but they modify by help by the scientists through the help of AI. So this the deep learning that use, for example, like deep BGC, Gecko, Satisfy. So this all the machine learning or the deep learning that we can use, to play around. So next slide, please. So the second one, so the using of AI in natural product research is for the structural characterization of natural product. So we can use AI, as a part of the prediction for the structure. So before this AI come across to this field, some compound that isolated, we cannot, maybe because of the low yield, we cannot characterize the structure. We cannot characterize the structure, or because of the limitation of the instrument that we use, yeah. So it's difficult for us to characterize the structure. Okay, so with AI, we can conduct the characterization. Yeah. So okay. So for conventional characterization, so these all the technique that we use. Yeah, all the technique that we use. Yeah, so we use an NMR. From NMR, we know the number of proton, the kind of proton and carbon. Yeah, so from IR, we know the functional group. From UV, we know the chromophore. Yeah, and then so from here, we some compound we can elucidate the structure. Okay, so after that, yeah, so with AI, yeah, so we there is the system that we can use, yeah. Okay, so we combine all the data that we get from the spectroscopy, yeah, spectroscopy. Yeah. Okay. So from the spectroscopy here. Okay. So we got all the methodological data. Yeah. So and then we can use, yeah, this computational data, yeah, to predict. Okay. So and then we can elucidate the structure, yeah, using the combination of the methodological data with AI. Yeah. Okay. Next, please. Okay. So this how, yeah, the MS, yeah, and NMR analysis that we can use, yeah, to predict or to elucidate the structure. Okay. So from MS we will get, yeah, the molecular weight, yeah, and then also the fragmentation of the compound. Yeah, the fragmentation. For fragmentation, we will see from the biggest, yeah, structure, they will fragmented into the chunk. So if you see here, so this the chunk for each chunk for the biggest structure. Okay, so after that, from NMR, yeah, we will have the proton NMR spectra. We will get the carbon NMR spectra. Yeah, the type of proton, the type of carbon. Yeah, so we get. Yeah, so we try to match. Yeah, so this what I call this neural network just now. Yeah, so this the data that we input, yeah, from MS, and then also, yeah, so we match, yeah, the spectral data, yeah, with the compound in the database of the system, because AI, like I said, is a database. They have all the info regarding all the structure. Like I said, yeah, AI is data mining. Yeah, so the data mining, you can imagine the number of compound in their mining. Yeah, start from the simple, yes, where chemistry started until now. Yeah, from the simple structure until the big structure. Big structure is, for example, like enzyme, protein. This is a bigger structure. Okay, so like I said, AI is data mining. So so from here, because they have all the structure, yeah, they have all the info. Yeah, when we put this input, yeah, to the system or to the deep learning, there is a process. They will process. Oh, this data, or this kind of MS they found in the structure, or this kind of the fragmentation they found in this structure. Yeah, so this what we call as the process in the neural network. Yeah, so they predict. Okay, so and then in NMR, when we put all the data NMR from our compound, yeah, so they also do the predicting. Yeah, from the mining. Yeah, from the mining. Yeah, so there are several deep learning that we can use. Yeah, for example, like this. yeah, until and then also the smiles. Yeah, smile is also the system, computational system that will generate the smile will generate the structure. Yeah, will generate the structure based on the mining that they have by the AI. Okay, so yeah, so this the tool of they integrate the tool of MS and then also NMR to predict the structure or for the structure characterization. Okay, so the next please. Okay, so now yeah, AI is used as a data mining to interpret the structure. The next one, to predict the target. Yeah, to predict the target. Okay, so the predict the target is more on the bioactivity. Whatever bioactivity that we want to test. Yeah, so remember AI is data mining. Yeah, AI is data mining. Okay, okay. So the predicting here. Yeah, so this to the target. What is our target? For example, as analgesic. Yeah. So what is our target? Yeah. So what is the enzyme for our target? Yeah. So we can use to predict. Yeah. So we just play around with the structure that we got from the prediction and using the software, you see is there any interaction between the small molecule that we discover with the target? Okay, so this the target, yeah, and then we also can see the biological activity, and also the toxicity, the toxicity. So we can predict using AI. Okay, so for this one, this to prioritize the area of chemical space, as with the high pharmaceutical potential, so for this one, so this is just a guideline. Okay, so and then the challenges for this for to target the predict the target and the bioactivity. So sometime when we maybe this compound that we isolate is a new or novel compound. Sometimes the novel compound, sometimes we don't know what is the target. So meaning that we need to do research slowly to see what happened. Okay, so here, so with the lag, here, so this will slow down the development, especially on the preclinical. Preclinical we use the animal model. Okay, so this one, so if we don't know the target, so this become a challenging for us to even we just use the AI, but if we don't know the target, so this will slow down the process. So this one, okay, so this the one of the challenges, but sometimes, because of this problem, some scientists try to develop the new development of AI, so they can discover this one. So they can give a rapid prediction, so with the AI. Okay, so this the AI tool, that usually use, maybe you recognize the molecular docking. Molecular docking, chemical clustering, so this is the technique that we can use, for the prediction. Okay, so next, please. Okay, so this one, to predict the bioactivity, so there are two, classical and chemoinformatic. What is the chemoinformatic? Computational and algorithm. So this is what we call as chemoinformatic. The combination of computational algorithm is more on the statistical. So when we combine, so this one what we call as chemometric approaches, chemoinformatic. Okay, so this tool like pass and see, yeah, to select pass and see, pass and see. Okay, so so this one, this actually this tool that have been adapted, yeah, for the natural product bioactivity prediction. Okay, so the challenging, yeah, so still there's a challenging for this method, yeah. Okay, so sometime, yeah, the natural compound, they're quite specific. Yeah, this are different with the synthetic compound. Yeah, so so this become challenging for when we start using this technique. Yeah, so for this situation, yeah, so the AI suggested to there is a pre-processing step. Yeah, pre-processing step. Okay, so to see, yeah, to see in terms of the chemical structure, yeah, the chemical structure with our target. Okay, so the next one, specialized, yeah, and a deep learning approach, yeah. So this quite a lot of approach or model that have been introduced, yeah, like as by the 3D pharmacophore. Pharmacophore is the functional group that responsible to give the action. Yeah? So meaning that this 3D pharmacophore will provide us what is the functional group in that structure that produce the action. Okay, so and then ChemProp, yeah, so this the another technique that have been introduced, yeah, and then this have been used actually, yeah, for predict the antibacterial activity of some compound, yeah, for example hylasin and so on. Okay, so this have been used. Okay, next. Okay, so here, yeah, so from the predicting biological activity, yeah, so we got everything, yeah, so our compound, our new compound, yeah, and then we predict the structure, we predict everything, yeah, and then we also have predict the bioactivity here. Okay, so after that, yeah, so from here. Okay, so from here, okay, so we will get yeah, from the compound we know, yeah, the gene, yeah, that responsible for synthesize the target molecule, yeah. So there is a genome feature that produced in the by the from the AI. There is a chemical feature, yeah. There is a biological feature. Yeah, so this will produce the this data in AI. Yeah, so with this data, yeah, so this data will be processed, and then we know, yeah, what is the action of this compound to that will produce the biological activity. Okay, so next, please. Okay, so from this this chemical feature, yeah, this chemical feature that produced, yeah, during the in the AI, yeah, from this chemical feature, yeah, so so this the metric, yeah, the metric that we got, yeah, from this metric, so we know, yeah, all the info regarding the chemical in terms of the structure, in terms of the modification that we can use, maybe if we conduct modification. So this will give a different action, whether the action may be increased or reduced. So this will from here, yeah, and then what is the macromolecule that can give interact, yeah, with this chemical. So this one actually, yeah, so this what we call as the chemical feature model, and then this from here, so we can I mean we have this all data, So if we need to synthesize, we can synthesize the drug. So maybe this in the nature they not produce this compound because we just play around with the software or system. So we need to synthesize. We synthesize and then we have the drug. So this drug is ready for the next process. Okay. Next slide, please. Next slide. Okay. So here, okay. So this all the database, for the natural product data. A lot of the database that we can use. How much the compound that have been isolated. From the natural resources. So this all the database. So we can utilize this. Bring this data set. To our study, so we can get a clear picture for our compound, for our drug candidate to produce a new drug. Okay, so next, please. Okay, so this continuation. So the next. Okay, so next. Okay, so this one, I just share my this one is my research when utilizing the chemoinformative. Okay, so my research background is more on the natural product, and then now I focus on the extraction. Extraction here because I more focus on the known compound. So how I can extract this known compound? So I can produce a high percentage of compound in the extract. And then this we can use maybe to produce the nutraceutical or cosmeceutical. So one of our research is on the utilizing of algae. Macroalgae, for the cosmetic formulation. Okay, so in this research we use two systems. RSM or response surface methodology, and then ANN. ANN is the deep learning. Deep learning. So artificial neural network. Why they call as artificial neural network? Because they adapt with our neuron, or natural neural network. Okay, so from the ANN, we this based on the prediction. Prediction. So we will for example like I focus on the extraction of fucoxanthin. So this carotenoid that present in macroalgae. Okay, so here I will gather all the information regarding the extraction of fucoxanthin. What is the parameter that maybe influence the extraction of the fucoxanthin? Maybe the temperature, the solvent, the type of extraction, and so on. Maybe also particle size of the sample. So we use ANN to see whether this actually will give effect on the percentage of fucoxanthin when we extract from the plants. Okay, so the data here, so we input the data to ANN, and then the ANN will process to see whether so all the parameter that we input here really affect the concentration of fucoxanthin after the extraction. So our output should be higher concentration of fucoxanthin. So we will see here. Okay, so and then after we see from this part, to see we will know what is the parameter actually responsible for the extraction of fucoxanthin. So we need to optimize. right? So we need to optimize. So the purpose of optimize to reduce the cost, yeah, to reduce the cost, especially when we work with the industry. Okay, so for optimization, we use RSM, response surface methodology. Okay, so here, yeah, there are the parameter that we get from here. Yeah, so we optimize here. Yeah, we optimize here. So we will get, yeah, the optimized condition to extract the fucoxanthin. So this what we do in our lab. Yeah, so to utilize AI together with RSM. RSM is not AI. Yeah, so RSM is more on the statistical analysis. Yeah. Okay, next, please. Yeah. Okay. Okay. So this actually AI work. Yeah. So this the input. Yeah, and then our brain. Yeah, there is a dendrite, there is axon, right? So so dendrite we receive, yeah, the input. Yeah, so and then this somata, yeah, where this will digest all the input, and then this the output. Yeah, so actually this adapt our neural system. Next, please. Okay, so this is what I said. Yeah, so this our neuron. Yeah, so the dendrite that will receive, yeah, all the info. So this the nucleus, yeah, or soma cell will digest all the information, and axon will pass this info to the next neuron as output. What is the reaction to our body? Yeah, so this is similar to the artificial neuron. Yeah, so this all the input. Yeah, and then there is a in the system that will digest, and then this the output. Okay, next, please. Okay, so this what I said that what we conduct in our lab. So we need to do this one. Yeah, so we need to do all the data preparation, all the data prediction. Yeah, and some experimentation. Yeah, so we will get all the model testing, yeah, and then we need to train the model. To train model, we use the RSM, yeah. To train here, we optimize, yeah. So for optimization, we need to design our design of experiment, yeah, design of experiment. So we will have the model of the output that produced from ANN, yeah, and then we will get the optimal solution. Yeah, or we will get the optimal solution. So actually this this one just for extraction. We can do this for formulation, for biological activity, for everything in a pharmaceutical field. Yeah, so we will shorten the process of research and then get the fast result. From this method. Yeah, so AI here is the tool that will help us. Don't let AI control us. We need to control AI. How we control? To utilize with this, so that we can make our life more easier in research. Yeah, by utilizing it. Okay, I think that's all for my presentation. Okay. Thank you.
We will be opening the question and answer session. If there's any question, please. Okay. Hi, hello. What are available AI online that can help in thesis for natural products discovery? What are available AI online that can help in thesis for natural products discovery?
For database AI, yeah, that free. Some AI is free, but some no. Yeah, sometimes the free one, yeah. So this not, yeah, you know, yeah. If free, this not, we cannot utilize. Yeah, it's quite limited. Yeah. So I think it's good if university can invest for this one. Yeah. So some university they invest on the AI, especially for the database, so we can use. Yeah, but I forgot. Yeah, there is one of the database that free. I forgot the name, so I will pass the name later on. There is especially for the nature product. Yeah, there is a free database. I forgot the name of the database. I will pass to you later. Okay. Okay. Yeah. Okay.
Thank you so much, Professor Dennis Susanti, for sharing today's material. For our next agenda, we'll be sharing materials which will be given by Ms. Maria Concepcion Sim, Magister of Pharmacy and Register of Pharmacy from Trinity University of Asia. Ms. Maria Concepcion Sim, Register Pharmacy and Magister Pharmacy, is a seasoned licensed pharmacist and academic leader with over 15 years of experience in pharmacy education, research coordination, and international academic collaboration. Currently serving as program chairperson of Pharmacy Department at Trinity University of Asia. She has held various academic and administrative roles across multiple institutions, including Our Lady Fatima University and St. Louis University. Her educational backgrounds includes a Master of Science in Pharmacy with a focus on manufacturing pharmacy and an ongoing doctoral research in public health. Ms. Sim is actively engaged in international academic initiative, serving as an adjunct, lecturer, and guest speaker in Indonesia, and participating in global conferences such as FAPA 2024 in Seoul. She also contributes in institutional development as chairman of the IACUC and a PAASCU accreditor, demonstrating her commitment to advancing pharmacy practice, education, and community health. We are honored to have Ms. Maria Concepcion Sim, who will be delivering today's lecture on characterization and antibacterial assessment of Mimosa pudica, or Macahia, PVA-based hydrogel against S. aureus. Ms. Maria Concepcion Sim, the floor is yours.
Yes, okay. Thank you very much. Hi. Okay, so how should I proceed with my PowerPoint? PowerPoint. Okay, so that is the so in the interest of time and in the interest of our stomachs for lunch, this will be very fast. This is the first and the last slide that I will present. Okay? Is that good? Okay. But they are not laughing. So only the Filipinos are only laughing on my joke. Okay, so it is my pride and privilege of presenting the study of one of our student researchers. Actually, they are here. Please stand up to be recognized. Ms. Lakap and Ms. Ornedo. So they are our student researchers, and as their co-advisor, I would like to present their study. So I was listening to the three discussions a while ago, and I would say this is the end point of what was being discussed. I mean, from the previous lectures, this could be the end product, because I was also would try to associate that, like Professor Tahir mentioned about the implementation on how to make these natural products be tangible, because that's the problem in research. We don't utilize. So that's our challenge. We keep on researching, we keep on discovering, but the most crucial part that I would say, and I believe that it could be the utilization. So on our end, in the Philippines, actually, in our university, we would still like to strengthen our research. And the genuine intent of this presentation is also to support further development of this study. So with the help of our partners here in Pancasila and our future partner in IIUM. So I was able to talk to Professor Tahir that I hope you will help us on developing the research. So to start with, the title of the research is the characterization and antibacterial assessment of Mimosa pudica. In the Philippines, we call that as makahiya. PVA or polyvinyl-based hydrogel against Staphylococcus aureus. So as we all know, skin diseases are being treated by topical treatment. So we cover our burns or our wound by simply applying bandages, okay? So however, there are limitations on using this conventional treatment in skin diseases. So there are very limited only for dry wounds. And infection could also be at risk. Scab formation and stickiness because the adhesiveness of different preparations available in the market could vary. And also non-selective debridement. It means to say if you use a conventional skin treatment, it could destroy both healthy and the infected skin tissues. So with the problem arising from the skin diseases, let's say for example for burn wounds, it could possibly develop into an infection, and the primary treatment that we use is only using a bandage or a gauze. So that's why, since also like I keep on mentioning that Professor Tahir mentioned about the optimization of the different preparations, might as well we would like to discover the possibility of this plant to be formulated as a hydrogel. So this is the I guess you are familiar with the patch. We use that, or simply your pimple patch, right? Our students in Trinity, you can see them like constellation, connecting all the dots while attending our classes. They have different designs. So that's why sometimes I used to, okay, I will ask a question from anyone who has a pimple patch with color red. Then they're going to start to peel the patch, okay? So that's why that's our inspiration. Now, as what we are also struggling with the plants that we have in our countries, actually, makahiya is found in both Philippines and in Indonesia, because we would like to discover a plant that is both found in, probably also in Malaysia. Professor Tahir, is there, what do you call that? Putri malu? Yes, yes, I just Googled that yesterday. Okay, so Putri malu, Putri means princess and malu means shy. So that's shy princess. Now in the Philippines, it's makahiya because hiya means shy. Now, since we have also a delegate from Thailand, I also Googled it. So it is mai, I don't know how to pronounce that, or yarap, it means touch me not. Okay, so, or in English, that is shame plant. So once you, I believe you know that, okay? So it's a weed in the Philippines. Also here, I mean also in Asian countries. So the takeaway that I got from Professor Tahir, Asian countries are green countries. It means to say we have a vast of plants that we can utilize. And so we can develop into an alternative treatment that will not be using synthetic materials that are toxic. Now, I was reading, yes, actually I just finished the PowerPoint yesterday. Okay? The students finished the study for a year, but I finished the study yesterday. Okay? So I was looking on what could be possibly be very same with the study of Makahiya in Indonesia and in the Philippines and in probably also in Malaysia, because I would like us to collaborate on how to make this product be patentable, because that's our challenge. We need to utilize. So for us to utilize, we have to make these researches to be in a tangible product. Okay? So when I was looking on the study in the Philippines, mostly it has a folkloric use that it has been used for dysentery or diarrhea. But I hope you can see the journal. But surprisingly, when I was looking on the journal, it is Indonesia who has the systematic review in Makahiya and not in the Philippines. So I need to walk out. No, okay? So it's surprisingly it is Indonesia that has a systematic review in Makahiya. So Dr. Denny should do this in the research. Okay? So they also conducted ethnobotanical of Makahiya. So I don't know if you can zoom that, or I hope you can see that, but I myself cannot see. Okay? But among all diseases, the topmost is in Indonesia, Makahiya is being used for skin diseases. In the Philippines, dysentery. But our students develop it into a topical treatment for skin diseases. So perhaps the students in Pancasila can furtherly develop and make us co-authors. Okay? Is that good? Yes. Okay? It's a good deal. Okay. Now, when I was looking on it's not a novel. I mean, it's not a novel study. That's why we go directly to the formulation itself. Now, when I was also looking on the journals, it is on the roots and on the leaves that have the active constituents responsible for its antibacterial property. And so the students use the parts of the Bakahiya. Okay, so I cannot really see that. So now, on the side of Makahiya, we already justified that it has a significant use as an antibacterial agent. Now we look on the other side, which is the delivery system, why we would like to formulate it as a PVA or a polyvinyl-based hydrogel and not as an ointment or not as a cream. Okay, so we would like to justify that a suitable dosage form is a polyvinyl alcohol because of its biocompatibility. You can just read on that. No, like what I've said, in the interest of time, I need to make it faster. So biocompatibility, safety, biodegradability, and ideal for skin promotions. So basically, the use of hydrogel is a more precise, and I would say a very selective manner of treating the skin disease. Okay, so I would like to share how the students, they were third-year students who used this method. So perhaps from Pancasila can also adopt the protocol that they have used. Now they have collected, they have identified, and then they utilize the different concentrations. The different concentrations were based on the journal of crude extract. So the crude extract is 10, 20, 30, 40, and 50. They had a journal for this for protocol. Then the process, they produce with their bare hands a hydrogel formulation or hydrogel base. And then they study that on the organoleptic, pH, and viscosity, physicochemical analysis, scanning electron microscopy or SEM, at which they examine that under electron microscope, and then after that, they use a software image J, which Professor Denny mentioned about the algorithm. So I suppose that the image J, if I'm correct, can be utilized as an AI algorithm, but it is not an AI. It is just being utilized as an algorithm. And also the FTIR. Okay. Now in the in vitro, we use Kirby-Bauer or this diffusion. And then the output, the desired output that we would like to have is an optimum. I would not say it's perfect because it's still a pre-formulation, but an optimum suitable product formulation as a hydrogen. Okay, so they collected the plants and then dried it and then ground. So I was asking them, why did you need to ground? Because for the undergraduate students, they usually use crude extract or ethanolic extract. So I asked them, why do you need to make it dried and make it granulated? So the purpose of drying the plant is to increase the surface tension. So if it's if the particles are smaller, the surface tension is high, and so the incorporation of the hydrogel will be facilitated. Okay, and it will be more soluble and also more stable. Okay, now the second part is they formulated the PVA. The PVA is ready-made already, but they make it as a 90% water and 10% PVA because polyvinyl alcohol is a water-based formulation. So that's why it's 90-10. So that's why the absorbability is also high. Okay, now they come out with 10%, 20%, 30%, and 40, 50%, and that is the mass. The rightmost is the mass of the macahiya extract, and the left side is the polyvinyl. Then we come out with the percent of the formulation. So for example, we have 15/30, that will make 50%. 10/25, that will make the 40%, and so forth. So looking on the table, I would say, I mean, you would like to predict that the most significant or the highest efficacy that is being formulated in hydrogel could be possibly the highest. Because when they were presenting it, I would know the end result. Most probably, the winner is the 50%. Okay, but let's see. Okay, now for the physical-chemical properties, we have the visual tactile method, pH measurement, because very important, the pH, so that it will be very compatible with the skin solubility and viscosity, and also the spreadability. Okay, so these are parameters that would also dictate or predict that the formulation is suitable. So the experimental hydrogen was being run also in FTIR, spectroscopy, and the hydrogel morphology, making it, or I mean, for them to justify really that the hydrogel is, and the macahiya extract can be blended uniformly, with uniformity. So in the in vitro, it's modified Kirby-Bauer. So again, I asked the students, why modified Kirby-Bauer? Because we used to use, you have finished microbiology, right? We are only utilizing Kirby-Bauer. However, since the extract is semi-liquid, we cannot place that in the disc because it will dry out, or it may be having a possible false positive result. So what they did is the semi-liquid methanolic extract formulated with the hydrogel, they submerged that in the, the disc was being submerged in the different concentrations, then they let it dry, and then place it in the agar. Okay? So that is the modified Kirby-Bauer. Now, the data analysis, the one-way ANOVA, normality or normal distribution rather, was also being identified, and the significance, significant difference. So let's now proceed to the results. So this is the pH of the extract, not yet the formulation. So the pH of the extract is slightly acidic, which we would say it's compatible with our skin because our skin is slightly acidic. So we know that also would facilitate also the absorption of the active ingredient. Now, the extract was being tested with different solvent to identify if the Macahiya extract would have a varying solubility in the three solvent system. So water, ether, and chloroform. So it's very fortunate that the Macahiya extract is highly soluble in water among the three solvents. So is it good? Yes, because the PVA is water-based. And so we have the rule of the thumb, like dissolves like. So if the extract is soluble and water soluble and the hydrogen is water-based, so we don't have a problem with the combination of the two systems, okay? Or of the two phases, okay? Now these are the samples of their formulation. So it has a direct relationship. As you increase the concentration, it's getting darker. Okay. Now for the pH measurement, still indeed, even that we have incorporated the extract with the PVA base, still it maintained the ideal pH range. So pH parameter, check, passed. Okay. Next. In the viscosity, this is the time that I was asking them during their defense in the viscosity. So we can see that among all, obviously, as the higher concentration, as the concentration increases, the viscosity decreases. Okay? So this trend suggests that addition of Macahiya extract interferes with the polymer network density of cross-linking efficiency, resulting in lower resistance to flow and therefore lower viscosity. So that's the problem, that's the arising problem during their defense when they were presenting it. Because in a normal or in a conventional definition, as the concentration increases, the efficacy also increases, right? So if we're going to look in general, we would like that the formulation would be having the highest extract concentration. But the problem is, with the highest concentration of the plant, it's becoming less viscous, which is not good. So they were now starting to cry that time. Okay? But I told them, don't panic, let's see. Okay? So, in the spreadability, so in the viscosity, we cannot decide yet which concentration is the most suitable. But when we look on the spreadability, yes, practically the highest spreadability we would expect from the pure, which is PMH0, pure, that is only PVA. So we expect that that is the highest. Now we're going to look on the constant on the mean. Second to the spreadability is that 10%. Okay? Now, what's the third runner-up? Who's the third runner-up? Most probably the 30. Can you see that? The third runner-up is the 30%. So I told them, okay, I can now see the picture. The candidate for the hydrogel is the 30% because it has a good spreadability and good viscosity. But the next question is, how about its significant difference on its antibacterial effect? Okay, now we have to go back to that issue. Now, to make it short for the hydrogel morphology, the hydrogel should be more porous. So if it is more porous, the absorption of the active ingredients is more exhibited. Okay? So to make it simpler, that's actually the SEM, or the scanning electron microscopy. Then after that, they use a software, the image J, to identify the porosity of the different concentrations. So this is the result. So what is our desired porosity? So among all, again, obviously, the most porous is obviously the pure. What do we like in the hydrogel? More porous, more viscous, and more spreadable. Okay? Now, obviously, the pure hydrogel is the most spreadable. But now with the concentration of the macahiya extract, the second runner-up is the 10. Okay? However, obviously, the 10% would not be having a good zone of inhibition or effect in the bacteria. So, delete 10%. Never mind. Okay? Next. Oh, it might be the 20%. But again, obviously, 20% might be only having a zone of inhibition, but not as strong as the higher concentration. So, again, the prediction, it can be the 20 or it can be the 30, but not really the 40 and 50. Why? Again, the viscosity is lesser. So the candidates are now the 30 and the 40, or perhaps the 20. So we have 3 candidates in the pageant. Okay? So next. Now in the comparative analysis, this is the height, the skewness, because they use the image J. So again, to make it shorter, we can see that in the 30, how should I use this? Anyway, so in the 30, you can see the 252, and in the 40, we have the 253. So eliminated 20%. The candidate now is remaining for the 30 and for the 40. Okay? However, again, the crucial, the higher the concentration, the viscosity is lesser. So now you know who is the winner. The 30. Okay, however, the question is, will it be good? Will it be a good antibacterial concentration? Okay? Because in the hydrogel formulation, it passed. The next question is its efficacy as an antibacterial agent. Okay? Now, this is the FTIR illustrating that the PVA hydrogel, even it is incorporated, even that the macahiya extract is being incorporated in the PVA, the active constituents that are responsible for the antibacterial are still present. So it means the formulation has not given a good effect, or it will not interfere in the delivery or exhibition of the active ingredients. So this testifies that the formulation is suitable for the macahiya extract. All right? So I have few minutes left. Now, we now found out that it's the 30% that is the winner for the hydrogel. But the question is, will it be effective? Now, I told them, okay, let's see. All right? So for the mean, the highest, the most effective antibacteria is the 50. But again, cannot be used because it failed the hydrogel parameters. So now the candidate will be the 30 and the 40. Now I asked them, let's see on your ANOVA. So I told them, I'm not a statistician, but I was able to interpret that or you look on the 30 and the 40. If there is no significant difference between the antibacterial property of the 30% and of the 40%, then might as well use the 30%. So the winner indeed is the 30% because the effect of the 30 and the 40 is basically the same because there's no significant difference. But between the two, the better hydrogen formulation is the 30%. And so we come out with the final product. So how should I, that one? That's the 30% Macahiya extract hydrogel formulation. Good viscosity, good porosity, good spreadability, and a good antibacterial effect that is comparable to a high percentage concentration of the Macahiya extract as an antibacterial agent. Okay? So I hope that the faculty of Pancasila and IIUM will help us support to develop the formulation. Perhaps you may start doing other than wound heal. Other than the pre-formulation and antibacteria, maybe you explore on the wound healing, anti-inflammatory, or other topical. Or perhaps you can develop a pimple patch out of it because pimple is stuff our use, right? This time, you should wash your face with Macahiya extract. Okay? So, that's also for future, for the recommendation. I hope we can optimize because in the ethnobotanical study of Indonesia, they use Macahiya with other plant extraction. So, they use multiple plant other than Macahiya. Or perhaps we can have a synergistic effect with active pharmaceutical ingredient. For example, ampicillin. Maybe we can incorporate ampicillin with Macahiya extract. All right? Advanced drug delivery application. Maybe the pH spreadability can be more enhanced. Integration of nanotechnology, sustainability, because we did not perform stability testing. You might explore the stab test. It can be accelerated stability testing because we don't know the storage condition of the hydrogel. And regulatory and safety considerations, maybe you can see, or you can, since we have used in vitro, perhaps you would like to discover it in vivo and explore the acute toxicity in the skin of the formulation. And regulatory, in the product registration, maybe we have different policies in product registration, but again, I hope this can be intangible product. I hope we can really develop the product. That's why I really present it to this platform for us to be supported by our university partners. So these are the references. See, I told you I will be fast. Okay. Yes. And then these are the researchers. May I again acknowledge our student researchers, Jaila Ferian Lagahid, Alicia Mathea Lakap, Dian Althea Gomuro, Tricia Hart Ornedo. The advisors, Mr. John Mark Soriano was here last year, and me as their co-advisor. Maraming salamat. Thank you very much for listening. If you have questions, you ask them. Thank you.
Okay, we would like to open the question and answer session. So, if you have any questions to Miss the session.
Okay, thank you. Ang dami.
Okay.
Okay, thank you.
My name is Siti Rayana. I'm from Universitas Pancasila, and my question is, what were the main challenges you encountered during the synthesis and characterization of the Macahiya PVA hydrogel?
Okay,
What
may
do you
I, all
challenge?
right. Miss Ornedo, please come here because they are the ones who really perform. I only presented it, but they are the performers. I am only the presenter. So yes, you can stay there.
So I think the question was the experiences, yes. During the formulation, the most hardest part was to find the appropriate time since we're still doing our studies for the SEM. And then the majority would be the formulation part in which we integrated the PVA and the Makaheya because truthfully, it was supposed to be a solid. So it would be proper for the Kirby Bauer method. However, the final product became a semi-liquid consistency. So that's it. We anticipated another form of the product, but we actually made something na, oh no. We actually made something more cohesive to put on the skin. That's it. Thank you. Hope I answered the question right.
Okay, thank you so much, Miss Maria Concepcion Sim, for sharing today's material. The next agenda is presentation of certificate, which will be given by Apotheker Gumilar Adi Nugroho as Farm Magister Science Herb. Please come up to the stage. Now, we would like to invite Professor Denny Susanti to come up to the stage, and Professor Taher to come up to the stage, and Miss Maria Concepcion Sim to come up to the stage. For the documentation committee, please take picture and 3, 2. Okay. Okay, for the documentation committee, please take picture in 3, 2, 1. Once again, 3, 2, 1. Okay, please take picture in 3, 2, 1. Once again, 3, 2, 1. Okay, take picture. The documentation committee. 3, 2, 1. Once again, 3, 2, 1. Okay, thank you. Please take your seat at the designated place.
So, before we move on to our next agenda, attendees are invited to take a break for lunch and to pray for our Muslim guests. Our break session is scheduled from 12 p.m. to 1:15 p.m. Once again, our break session is scheduled from 12 p.m. to 1:15 p.m.
Cek, cek. Oke, buat teman-teman silakan langsung ambil makan siang ya. Makan siangnya di sebelah sana. And for Philippine friends, it's a lunch time. You can take your lunch at there. So, thank you.
Yes. Attentions to all participants. Our break will end in 5 minutes. Attentions to all participants. Please kindly enter the room because we'll start in 5 minutes. Attentions to all participants. Please kindly enter the room because we'll start right now. Once again, attentions to all participants. Please kindly enter the room because we'll start right now.
For our next agenda, we'll be sharing materials which will be given by Professor Dr. Rarnath Apotheker Dehni Rahmat, Magister of Science from Fakultas Farmasi Universitas Pancasila. Professor Dr. Rarnath Apotheker Dehni Rahmat, Magister of Science, is a distinguished academic and pharmaceutical scientist specializing in nanotechnology. Born in Garut on December 27, 1978, he currently serves as professor and head of doctoral program in pharmaceutical science at Universitas Pancasila. His academic journey spans multiple prestigious institutions, including ITB, Innsbruck University, and NTU, with expertise in pharmaceutical nanotechnology and molecular oncology. He has delivered invited lectures globally, secured numerous national and international research grants, and earned various accolades, including innovation awards and patents in nanomedicine. With a Scopus H index of 16, he continues to contribute actively to research, education, and public engagement in pharmaceutical science. We are honored to have Professor Denny, who will be delivering today's lecture on nanotechnology on the development of herbal medicine, nano beauty. Professor Dr. Renat Apotheker Denny Rahmat, the floor is yours.
Thank you so much. So I would like present a topic associated with nanotechnology. Okay, please share my presentation. Okay, I think, okay, yeah. My topic is about nutri-cosmetics, the development of nanocosmetic, nanotechnology for Putri, Putri Indonesia, you know? Or Miss Indonesia, you know? How about Bini Bining? No? How about Philippine student? You know Bini Bining? Okay. Also, Bini Bining is like a beauty pageant, right? Yes, yes, yeah. Okay. Yes. For Philippine student, just quiet because I will show you and I will ask Indonesian student to guess who is she. Okay, next slide. Oh. I think Philippine student knows her, right? How about Indonesian student, you know? No. Okay. I will ask Philippine student, who is she?
Is Atiza Manal.
100% right. Yeah. So Atisa Manalo is like a beauty queen, like maybe like in Indonesia. You know who is Putri Indonesia in Indonesia? Angelina Sonda, yeah, you know, yeah. So we can learn from her because she found, she tried her best to be Miss Universe Philippines. Yeah, she fell in 2018, but somehow she competed in Miss International in Japan. But she fell again. Yes, and also in 2024, she tried again in Bini Bining. Yes, but fail again, but somehow she sent to Cosmo, Miss Cosmo. Who is Miss Cosmo from Indonesia? You know? Who is Miss Cosmo from Indonesia? You know Miss Cosmo? You know? Tata Juliarti. You know? Tata. Who?
Julia Street.
100% right. Yes. But somehow she also failed, yeah, in that competition, and Tata Julia Street won, yeah, Miss Cosmo. Now she is in Vietnam, I think, yeah. But she keep going, yeah, try again, and the winner of Miss Universe Philippines, right? Yeah. She won. Hello. Okay. Last year, Miss Connie, we met, yeah. And then today we meet again. Also, Miss Krista. And new members, faculty. Yeah. Oh, last year, yeah. Okay, welcome in Indonesia. Thank you so much, yeah. For the second time, Philippine students join the summer course, yeah. Especially you meet again. Hello. Where is Aldo? You don't know? Okay, yeah. Okay. We should learn from her because, I mean, even she failed, she tried again and again. Finally, eventually, she won. But, you know, if we notice the year from 2018 to 2025, her face is still the same, even younger right now. Yes, even younger. Of course, I mean, she has tried her best, I mean, to protect her skin, yeah, to consume supplements, yeah, to keep her beauty. Okay, next. Oh, I will ask Indonesian student, who is blondie hair? Who is with the blondie hair? Oh, 100%, right? Who is she? Beauty influencer, yeah. She is very nice, yeah. Always, I mean, accept endorsed from various small industries, cosmetic industries, various small brand cosmetics even. She try many times, I think every day. But her skin still okay. Yeah. She's beautiful, she's very nice. So, I mean, it's not about sunlight, also oxidant, aging, wrinkle, acne, but also daily activities, behaviors. So, I mean, if your activities is risk, yeah, against the condition, environment, harassment environment, yeah, that will destroy your face, your skin, then you should put your effort more, yeah. I think Tasha, yeah, also try her best, yeah, to protect her skin because every day she try many times, yeah, with the cosmetic brands. And who is the girl with long hair, black long hair? Who? Who answered the question? I heard somewhere. Oh? Who answered the question?
Harasta Haifa Zahra.
Why do you know? Why do you know? Yes, she is also like Artista Manolo, but Indonesia, yeah. But she won. She won Miss Supra International, Supranational, yeah, in Polandia, yeah, I think. That's why she traveled a lot, yeah. She traveled a lot and come to one country to another country, yeah. I think every month, yeah. To give a crown for the new queen, yeah. And I think because her activities, yeah, from country to another country again and again, every month, also she try her best, yeah, to protect her skin. Next. Otherwise, I think you will have problems, yeah, in your skin, yeah. Even your skin will expose you to the suns, yeah, to the sun rays, yeah, that can cause cancer. Now we have here nano cosmetic. It means that the ingredient for cosmetic that we can modify into nanoparticle. Yeah, especially for active ingredient. Because right now, back to nature is like trending, yeah. Then the cosmetic contain natural products, yeah. Okay. Why should it be transformed into nanoparticle? Of course, yeah, we can find advantages nanoparticle over the traditional one. Yeah. Okay. Here, with the introduction of nanotechnology into cosmetic, new era of sophisticated skin product that target different skin issue and enhance general skin health has begun. Because, you know, every nanoparticle, without nanoparticle, the active compound can go anywhere without purpose, without goal. But with the formulation of nanoparticle, the active compound can target into the specific site, even specific layer of the skin. Yeah. By knowing the characteristics of the target layer of the skin, yeah, then the nanoparticle can stay at the target. So it's important, I think, yeah, for you to use nanotechnology as a strategy, yeah, to target the specific site, yeah. Here, ingredients used in nanocosmetic are often measured at the nanoscale. So, nanoparticle is small, but how small? Yeah. Here, the nanoparticle for the cosmetic named nanocosmetic rank from 1 nanometer up to 100 nanometer. But in general, we can say that nanoscale is in the rank of 1 up to 1000 nanometer. But here smaller, yeah. Here smaller for nanocosmetic. We can define smaller than it should be. Even can accept, yeah, up to 1000 nanometer, but still, yeah, we can make it smaller, even much more smaller, yeah. The nanoparticle has various methods, has various components, yeah, that we can formulate, and we can use various ingredients, yeah, for the specific method and also for the specific type of the nanoparticle. Because by formulation of nanoparticle, the performance, distribution, and efficacy will be better. Yeah. So it's like you just go ahead, strike forward to the target without walking around. Then for the time, for the energy, yeah, for the concentration, yeah, for the focus, you will have a lot. You will obtain a lot. It means that efficacy, yeah. Next. So the advantage of the nanoparticle, this make it possible for chemical to penetrate the skin more deeply and reach layer that may be difficult for regular substance to reach, yeah. Because the small size can go anywhere, yeah. So I am bigger. I cannot go anywhere because heavy and yeah. But if something is small, that then can go even through the small space, like intracellular, yeah, between the cells. So the mechanism of the nanoparticle can go through the skin by passing through the small space, pores, yeah. Advanced delivery method, including nanoemulsion and nanocapsule, are made possible by nanotechnology. So one of the example is nanoemulsion, and the other is here we have nanocapsule. It's like capsule, but in small size. Yeah. How about nanoemulsion? It's also like macroemulsion. Yeah, we have big droplets of oil in water. Yeah, it could be like that. But also we can make it smaller, but with specific method, yeah, that can break big globule into very small size. Yeah, here we have the target. Yeah, of course, the skins, to ensure that active chemical reach the desired skin layer for optimal efficacy. Yeah, why we should apply nanotechnology or nanoparticle for the cosmetic? Because the efficacy will be better. Yeah, because some active compounds works in the layer of the skin, not on the surface of the skin. So it needs for the compounds for going through, for passing through the skin. Especially stratum corneum. Yeah, stratum corneum is like the upper layer, yeah, of the skins. Next, better penetration through the skin barrier is made possible by the smaller particle size of the material used in cosmetic, yeah. And for some cases, which is nutri-cosmetic, yeah. So in this case, the compound is delivered by oral administration. So nutri-cosmetic is about oral consumption. So you need not only skin application, yeah, application on the skin for the cosmetic, but also you need supplement that can nourish your skin. So it will be much better, yeah, for the effect. Then we can talk about the bioavailability. It means that the compound should be in the bloodstream, should be absorbed, yeah, into bloodstream, distribute, yeah, within the circulation in the body, and give the efficacy. And the effect will support, yeah, the health of the skin. Here also we can see specific skincare requirement can be addressed by tailoring nanocosmetic composition, yeah. Because you know you want this compound to be released in 10 minutes, in 30 minutes, even longer, yeah, continuously. So you should modify the nanoparticle used for cosmetic, yeah. Next. So because of their adaptability, nanoparticle may be used to create customized solution that address a variety of problems, including pigmentation, hydration, aging, and more. So by the nanoparticle, we can overcome this problem, which are pigmentation. So your skin will get dark, yeah. And for maybe Asian people, we don't want to have dark skin, I think, yeah, especially for the girls or women, yeah. So I mean, pigmentation is about the aging, it's about the oxidation, so we can deliver active compound that has capacity for scavenging, of free radical can inhibit aging. Also, we can also deliver the compound that can absorb the water to moisturize, your skin. So the water content of the skin will be increased, and the regeneration of the cell will be okay, and you will have healthy skin. Next. So I will give you little bit example, of the natural product that we can use for cosmetic formulation. Such as here we have Centella acetica, here Centella asiatica, increase blood circulation. So circulation of the blood can be better, then it will deliver the nutrients, from the cosmetic to the dermis, Then the cell in the dermis will be okay, will be better, will be healthy, The degeneration will be fine, So the collagen synthesis will be improved, and tissue, also skin tissue, to stay youthful. So you can see Atisamanolo is, I think looks younger, than she is, I think. Because maybe she knew something, daily, or she consumes something daily, every day, to nourish her skin, to keep younger. I think you should do that, So, Centella acetica is good for collagen synthesis, and collagen play an important role in increasing skin elasticity, so that sign of aging such as wrinkle and fine lines do not occur too early. Okay, next. So we have also here asiaticocytes. It's good for treating sensitive, inflamed, irritated, and itchy skins. Sometimes, after using the cosmetic, so the skin is sensitive, you feel itchy, and then irritated. So to calm down your skin, you can also use the cosmetic containing asiaticocytes. Next. Here, others function of the pegagang, Centella asiatica. We can apply for many disorders, many problems. And fortunately also it will show antioxidant activity for aging, to inhibit the activity of free radicals. Free radical is dangerous because it's reactive, can react with the protein, with the collagen, DNA, which is important, for keeping your skin youthful. Next. So the others natural product we can use for the cosmetic is curcuma longa, but the color is yellow. We can use it as a sunscreen, but don't worry. That's why we should use nanotechnology strategy, nanotechnological strategy for masking Yeah, the yellow colors of curcuma longa. Even it's good, yeah, for the skin. Can moisturize. Also has antioxidant activity and sunscreen. Do you use sunscreen for your daily care? For your skincare? Yes? Really? Can you mention your product or cosmetic product? What is the brand of your cosmetic product for the sunscreen?
Laneige.
That what? Oh, Prada. My God, Prada. You know it? You know? You know. So mention again. So mention again the name. Maybe you can clearly name it. You can clear mention of the brand. So what is the brand for your skincare? Berapa kakak budget kakak untuk you can mention your brand of your skin.
Sunscreen. My sunscreen is Wardah.
Wardah.
Yeah. Wardah. The budget is maybe fifth. Oh iya, 45 berapa? 14,500 rupiah. Ya. Ya, not really lah.
How about you?
My skincare is Labore. Labore is from cosmetic from Indonesia.
Yes.
Budget?
The budget? Yeah, how much?
Oh.
Right here.
In 1 month, like 500K.
What? 100? 100?
500.
Five?
Yeah, for 1 month.
How about you?
I use Face Shop. It ranges from 500 to 1,500 pesos.
Is expensive? Oh, okay, good. Yeah, hopefully your skincare content centella asiatica or curcuma longa. Next slide, please. So the advantage of this natural product, yeah, can reduce itching, yeah, and also redness, swelling, yeah, because of the inflammation. Then several studies have shown that turmeric can help also reduce dark spots because pigmentation, yeah. And in fact, one study found that using turmeric extract for 4 weeks reduced dark spots by up to 14%. Next. The last, yeah, for the example, aloe vera, yeah, as an alternative in medicine, but also we can use for cosmetic. I think the well-known brand from Korea is Republic, yeah. It also contains so much concentration, yeah, high concentration of the aloe vera. Here, we can find aloe vera in many forms, yeah, like in capsule, powders, yeah, gel, of course, for the cosmetic, yeah. Next. Even we can eat aloe vera. We can make drink from aloe vera, and it's good. Yeah, we can find the drink in the supermarket, yeah, containing aloe vera, yeah, like small cubic, yeah. I think the taste is still okay, yeah. Little bit sweet is fine, and also we can put sugar if you want, but also sugar is not healthy, yeah. Next, then in my case, I study about a serum for cosmetic, yeah, containing curcuma longa, centella asiatica, and also aloe vera. And next, That's the publication. So, I mean, the formulation can show antioxidant activity, yeah, the serum. Hopefully also you can use serum for your cosmetic because it contains high concentration of the active compound, then it will show high activity, yeah, better efficacy. In here, we can find the high activity of antioxidant. Next, also, it's good for collagen, yeah, for collagen here, and also for the growth factor that governs periocellular. Oh my God. Please stop the lecture. Okay, fine. Because you will go in, I think, in around 15 minutes, right? Okay, don't worry. Yeah. Okay, so cellularly, I think it's good also for you to know the mechanism or the function of the natural product, yeah, how it works in the skin. Here, we have quite wow term, like here we COL, COL, or call, yeah, COL1A1 is known as the gene encoding the alpha 1 chain of the type 1 collagens, yeah. So we can find the gene that can express the protein, especially specific protein for the regulating the skin, regulating collagen, regulating here growth factor. That's good for the skin, to make your skin younger, yeah, look youthful. Here, for TGF beta 1, such as cell, for the function is cell proliferation, differentiation, ECM production. So you will not have wrinkle, yeah, fine lines in your face. Oh, you look old because you have wrinkle. So your chin is bigger than it should be. Then you can use natural product that can increase growth factor. Next, also each YAL1, yeah, which is hyaluronidase enzymes. So also in our body, especially in the skin, also we have enzymes that can destroy something good in the skin. For this case, hyaluronic acid is good for moisturizing your skin because it like absorbs the moisture, the water, so your skin will be hydrated. You look shiny, glowing, yeah. The water is good for regenerating the new cells, yeah. Also, we have here FGF2 is a fibroblast growth factor, yeah, that promotes cell proliferation. So we need the natural product that can increase the fibroblast growth factor, but not increase hyaluronidase enzymes. Yeah. We should reduce. And God delivers the natural product to reduce here hyaluronidase enzymes. Next. Yeah. Yeah, here we have COL, as I told you before, yeah, call 1A1 gene plays a role in collagen synthetic, a key component of the skin's extracellular matrix. Yeah, the matrix that can connect, yeah, one cell into another. Then you look your skin quite tight, yeah. That it will look good. Here also, several studies have shown that TGF beta 1 expression plays an essential role in regulating cell growth and collagen formulation. Yeah. Then from now you should find your cosmetic not just because of expensive, very well known brand, but from now you should search brand cosmetic that contain active compound that can increase, yeah, collagen synthesis, fibroblast growth factor, reduce enzyme that can destroy hyaluronic acid, yeah. Not just purchase and purchase and purchase, but you never try on your skin, yeah. Okay, next. Here we have, so now you should keep in your mind that your brand cosmetic contain active compound that upregulating at COL01A1, upregulating at TGF beta 1, FGF2, but downregulating at hyaluronidase 1 that reduce hyaluronic acid, yeah, degradation. So you should find something that can reduce degradation of hyaluronic acid. Next, for aging, we have here the effect, yeah, of free radical that react with protein, collagen, specific amino acid, DNA, yeah, that will destroy the skin, can cause cellular damage, decrease proliferative response, defective host defenses, yeah, due to the free radical resulting from exposure to the sunlight, sun ray, dirt area, yeah, inflammation, your stress, your boyfriend that make you stressful, you should kick out, yeah. Okay, then you will I think you will not have free radical and you will have good skin, very healthy. Because otherwise, I mean, if we don't have a free radical, then we will have hemostasis, then normal growth and metabolism occur, take place. Next slide. So keep in your mind negative rule, excessive proactive, yeah. Oxygen species can lead to oxidative stress, can contribute to the skin aging, worsen inflammation, impacted in the development of skin cancer. Yeah. So from right now, please consider your brand cosmetic that you use everyday, daily care. Yeah. Otherwise, your skin is getting worse. Next, as I mentioned, that we can apply nanotechnology to have the nanoparticle, then the cosmetic will be nano-cosmetic. Yeah, with the advantages over the traditional one. Here we can use liposome, nano-emulsion, nano encapsulation that can deliver, yeah, the active compound into the skins in the right position. Next, nanoparticle in sunscreen, yeah, we can use because, I mean, sometimes maybe you use sunscreen containing titanium oxide, zinc oxide, yeah, and if you apply too much on your skin, then your skin will be chalky, very white colors, yeah, and everybody will afraid of you. Then you should like choose nano particle cosmetic, yeah, nano cosmetic for zinc oxide, titanium oxide for sunscreen to have transparent effect. So even you use sunscreen containing zinc oxide, titanium oxide, then still your natural skin is glowing, yeah, without any effect from the white color of the zinc oxide, titanium oxide. Next, of course here, yeah, we can formulate nanoparticle into serum, cream, lotion, yeah, that contain vitamin, antioxidant, moisturizing agents, yeah. Natural product also, like curcuma longa has activity as moisturizer, yeah. These nanoscale structure are used in the creation of the sophisticated skincare solution that address certain issue, including skin restoration, hydrating, and anti-aging. Yeah, especially for this case that we can use nano cosmetic. Yeah, generally. Next. Finally, we are in the last section of this lecture. Wow. So you can go to solo. Okay, I have the nano cosmetic. Now I'm going and everything's fine. Yeah. So Nutri Cosmetic, oh sorry, it's from my handphone. Combination of the word nutrition and cosmetic refers to skincare, yeah, oral skincare that you consume orally by oral administration, yeah, to support your skin. Not only application of the brand, your brand cosmetic on the skin, but also consume orally. So consuming orally for the skincare, it's called Nutri Cosmetic. Yeah. Here, Nutri Cosmetic are ingested orally. So you need something to drink containing product, yeah, that is good for skin. Not always coffee, you know coffee, chocolate, and also Coke, matcha, acha matcha. My God, you know, yeah. But also you should drink something which is good for your skin. Wow. Korean boy band. Very glowing, shining, yeah. Because you daily consume Nutri Cosmetic, ingested product orally, yeah. Here, antioxidant like polyphenol, beta carotene, vitamin C, and E are frequently in Nutri Cosmetic to scavenge free radical and guard against oxidative stress. Next. Supplements containing collagen are often used to promote skin supplants, yeah, and lessen the visibility of wrinkle and fine lines. Yeah. Next. Yeah. Nano emulsion, as I mentioned before, containing oil droplet in water, can deliver the natural product. Yeah. Next. Because also nano emulsion have the potential to enable the regulated release of active compound. So why should we formulate natural product into nano emulsion? Because nano emulsion can release in regulation. Yeah. Next, nano encapsulation. So different from nano emulsion, nano encapsulation is like capsule that have shell to protect the active compound. So the active compound is protected by the capsule in nanoscale size, which is 1 up to 100 nanometers, because you think that your natural product is sensitive for degradation. Yeah, it's not stable, it's unstable. So you should use nano capsule to protect the active compound and also to release in regular way, in control release profile. Next, here we underline, emphasize, yeah, the function of the nano encapsulation for the control release, yeah, efficient release of active ingredient, providing a wide range of compatibility and efficacy in cosmetic composition. Next, conclusion. So you will never find again the new slide then. So for the conclusion, ingredient can be targeted to specific skin layer or cell by nano encapsulation, allowing for a more focused therapy of condition including wrinkle, fine line, pigmentation, or dehydration. Then ingredients that are unstable or sensitive are shielded against the destruction using nano encapsulation. Yeah, and also here nano cosmetic provide a more efficient and focused approach to skin care. That's all my presentation. Thank you so much. Maybe you have question, please. Yes.
I'm currently working on a thesis right now, and on a thesis, and my yes, and my idea may be using type 1 collagen to develop supramolecular collagen-based nanoparticles for anti-wrinkle effect. But since collagen type 1 alpha expression naturally declines with age, how would you recommend I optimize the formulation and delivery to enhance skin penetration?
Of collagen.
Yes, pa.
Thank you so much, yeah, for your very wow question. Unbelievable. It's very difficult to answer, and I think I need one night to answer. Please don't go away to Solo. Just stay here, then I will find the answer. No. Yeah? Okay. But so for the best formulation that that we can attempt for your formulation containing collagen is that, of course, yeah, nanoparticle. Yeah, because nanoparticle can make the deliver efficiently. For this case, we can use polymeric nanoparticle. Yeah, polymeric nanoparticle or etosome, yeah, which is what the type of nanosome, yeah, using vesicle that contain collagen composed of something that can modify the barrier properties of the skin. So the barrier properties of the skin will be better, easily to be penetrated, become more fluidity, and the nanoparticle will easily pass through the skin because of the composition in the nanosome. Nanosome can be liposome, can be neosome, can be ectosome, which has specific mechanism of the transport. Yeah, transferosome. Yeah, something som. It's good for your formulation because it can contain something that can modify the barrier properties of the skin. Yeah. I mean, if your boyfriend is very angry, very unfriendly, then you should buy something for them, for her, for him. Okay. It's like you make nano formulation that contain something that can lower the barrier properties of the skin. Okay? It's okay for you? And you?
A very curious question because you stated that the nanoparticles, when by oral consumption, it can bypass through deeper levels of the body. Bypass the deeper. Yes. Bypass. Yes, and I was just curious if it can be harmful if it crossed the blood-brain barrier.
Harmful. Oh, very good question. Very good question. Yeah, bypass the distribution and somehow it will reach the brain because it can penetrate blood-brain barrier. Yeah, that's why we should choose very proper composition. So in specific composition, it is possible to pass through the brain, but by passing through the blood-brain it's difficult. For specific composition and ratio of the weight of the formulas of the ingredient, so the nanoparticle will not reach brain, so it will be safe. So be careful to choose the composition of nanoparticle, the composition of the ingredient of nanoparticle. Be careful to choose your boyfriend. Okay? Do you have any other question? Or is it enough? Enough? Oh, okay. So have a nice travel safely to Solo. Okay? Thank you so much. Bye, everyone.
Thank you so much, Professor Denny, for sharing today's material.
To Prof. Denny, please stay in front of the stage because right after this, the agenda is the presentation of certificates that will be given by Apoteker Gumilar Adi Nugroho, Sarjana Farmasi Magister Science. Okay. For the documentation committee, please be ready. Okay, please proceed the picture in 3, 2, 1. Okay, once again, in 3, 2, 1. Okay, thank you. Please take your seat at the designated place.
By dart marks the end of our opening ceremony. Once again, best wishes to all participants in the summer course of 2025. We hope you enjoy our series of events and for upcoming days as well. My name is Azumi Sultanika, together with my partner,
Samai Fadimas.
we would like to say,
Thank you for attending this opening ceremony. Good afternoon and see you soon. Okay, but before that, attention to all participants. Do not leave the room yet because there will be an additional announcement. First, we will take you to go on the stage to take a picture together. So please come up to the stage to take a picture together. Okay, so for the participants, please go up to this, go onto the stage. For the lecturer, please in front of the stage. Okay, for participants, you can go up on the stage. Okay, so there is an announcement. So after this, all of you are going to the bus. And if you want to change your clothes, you can change in the rest area during the dinner time. Thank you.